Chapter 8. Molecular Biology and the Origin of Life. In previous sections of this book, we have shown how the fossil record invalidates the theory of evolution. In point of fact, there was no need for us to relate any of that, because the theory of evolution collapses long before one gets to any claims about the evidence of fossils. The subject that renders the theory meaningless from the very outset is the question of how life first appeared on Earth. When it addresses this question, evolutionary theory claims that life started with a cell that formed by chance. According to this scenario, 4 billion years ago various chemical compounds underwent a reaction in the primordial atmosphere on the Earth in which the effects of thunderbolts and atmospheric pressure led to the formation of the first living cell. The first thing that must be said is that the claim that non-living materials can come together to form life is an unscientific one that has not been verified by any experiment or observation. Life is only generated from life. Each living cell is formed by the replication of another cell. No one in the world has ever succeeded in forming a living cell by bringing inanimate materials together, not even in the most advanced laboratories. The theory of evolution claims that a living cell which cannot be produced even when all the power of the human intellect, knowledge and technology are brought to bear nevertheless managed to form by chance under primordial conditions on the Earth. In the following pages, we will examine why this claim is contrary to the most basic principles of science and reason. An example of the logic of chance: if one believes that a living cell can come into existence by chance, then there is nothing to prevent one from believing a similar story that we will relate below. It is the story of a town. One day, a lump of clay, pressed between the rocks in a barren land, becomes wet after it rains. The wet clay dries and hardens when the sun rises, and takes on a stiff, resistant form. Afterwards, these rocks, which also served as a mold, are somehow smashed into pieces, and then a neat, well-shaped, and strong brick appears. This brick waits under the same natural conditions for years for a similar brick to be formed. This goes on until hundreds and thousands of the same bricks have been formed in the same place. However, by chance, none of the bricks that were previously formed are damaged. Although exposed to storms, rain, wind, scorching sun, and freezing cold for thousands of years, the bricks do not crack, break up, or get dragged away, but wait there in the same place with the same determination for other bricks to form. When the number of bricks is adequate, they erect a building by being arranged sideways and on top of each other, having been randomly dragged along by the effects of natural conditions such as winds, storms, or tornadoes. Meanwhile, materials such as cement or soil mixtures form under natural conditions, with perfect timing, and creep between the bricks to clamp them to each other. While all this is happening, iron ore under the ground is shaped under natural conditions and lays the foundations of a building that is to be formed with these bricks. At the end of this process, a complete building rises with all its materials, carpentry, and installations intact. Of course, a building does not only consist of foundations, bricks, and cement. How, then, are the other missing materials to be obtained? The answer is simple. All kinds of materials that are needed for the construction of the building exist in the earth on which it is erected. Silicon for the glass, copper for the electric cables, iron for the columns, beams, water pipes, etc. all exist under the ground in abundant quantities. It takes only the skill of natural conditions to shape and place these materials inside the building. All the installations, carpentry, and accessories are placed among the bricks with the help of the blowing wind, rain, and earthquakes. Everything has gone so well that the bricks are arranged so as to leave the necessary window spaces as if they knew that something called glass would be formed later on by natural conditions. Moreover, they have not forgotten to leave some space to allow the installation of water, electricity and heating systems, which are also later to be formed by chance. Everything has gone so well that coincidences and natural conditions produce a perfect design. One who manages to sustain his belief in this story so far should have no trouble surmising how the town's other buildings, plants, highways, sidewalks, substructures, communications, and transportation systems came about. If he possesses technical knowledge and is fairly conversant with the subject, he can even write an extremely scientific book of a few volumes stating his theories about the evolutionary process of a sewage system and its uniformity with the present structures. He may well be honored with academic awards for his clever studies, and may consider himself a genius, shedding light on the nature of humanity. The theory of evolution, which claims that life came into existence by chance, is no less absurd than our story, for, with all its operational systems, and systems of communication, transportation and management, a cell is no less complex than a city. In his book Evolution, A Theory in Crisis, the molecular biologist Michael Denton discusses the complex structure of the cell. To grasp the reality of life as it has been revealed by molecular biology, we must magnify a cell 1,000 million times until it is 20 kilometers in diameter and resembles a giant airship large enough to cover a great city like London or New York. What we would then see would be an object of unparalleled complexity and adaptive design. On the surface of the cell we would see millions of openings, like the portholes of a vast spaceship, opening and closing to allow a continual stream of materials to flow in and out. If we were to enter one of these openings we would find ourselves in a world of supreme technology and bewildering complexity. Is it really credible that random processes could have constructed a reality, the smallest element of which a functional protein or gene is complex beyond our own creative capacities, a reality which is the very antithesis of chance, which excels in every sense anything produced by the intelligence of man? 238 The complex structure and systems in the cell. The complex structure of the living cell was unknown in Darwin's day and at the time. Ascribing life to coincidences and natural conditions was thought by evolutionists to be convincing enough. Darwin had proposed that the first cell could easily have formed in some warm little pond. 239 One of Darwin's supporters, the German biologist Ernst Haeckel, examined under the microscope a mixture of mud removed from the seabed by a research ship and claimed that this was a non-living substance that turned into a living one. This so-called mud that comes to life, known as Bathybius haeckelii, Haeckel's mud from the depths, is an indication of just how simple a thing life was thought to be by the founders of the theory of evolution. The technology of the 20th century has delved into the tiniest particles of life, and has revealed that the cell is one of the most complex systems mankind has ever confronted. Today we know that the cell contains power stations producing the energy to be used by the cell, factories manufacturing the enzymes and hormones essential for life, a databank where all the necessary information about all products to be produced is recorded, complex transportation systems and pipelines for carrying raw materials and products from one place to another, advanced laboratories and refineries for breaking down external raw materials into their usable parts, and specialized cell membrane proteins to control the incoming and outgoing materials. And these constitute only a small part of this amazingly complex system. W. H. Thorpe, an evolutionist scientist, acknowledges that the most elementary type of cell constitutes a mechanism unimaginably more complex than any machine yet thought up, let alone constructed, by man. 240 A cell is so complex that even the high level of technology attained today cannot produce one. No effort to create an artificial cell has ever met with success. Indeed, all attempts to do so have been abandoned. The theory of evolution claims that this system which mankind, with all the intelligence, knowledge and technology at its disposal, cannot succeed in reproducing came into existence by chance under the conditions of the primordial earth. Actually, the probability of forming a cell by chance is about the same as that of producing a perfect copy of a book following an explosion in a printing house. The English mathematician and astronomer Sir Fred Hoyle made a similar comparison in an interview published in Nature magazine on November 12, 1981. Although an evolutionist himself, Hoyle stated that the probability that higher life forms might have emerged in this way is comparable to the probability that a tornado sweeping through a junkyard might assemble a Boeing 747 from the materials therein. 241 This means that it is not possible for the cell to have come into being by chance, and therefore it must definitely have been created. One of the basic reasons why the theory of evolution cannot explain how the cell came into existence is the irreducible complexity in it. A living cell maintains itself with the harmonious cooperation of many organelles. If only one of these organelles fails to function, the cell cannot remain alive. The cell does not have the opportunity to wait for unconscious mechanisms like natural selection or mutation to permit it to develop. Thus, the first cell on Earth was necessarily a complete cell possessing all the required organelles and functions, and this definitely means that this cell had to have been created. The problem of the origin of proteins so much for the cell, but evolution fails even to account for the building blocks of a cell. The formation, under natural conditions, of just one single protein out of the thousands of complex protein molecules making up the cell is impossible. Proteins are giant molecules consisting of smaller units called amino acids that are arranged in a particular sequence in certain quantities and structures. These units constitute the building blocks of a living protein. The simplest protein is composed of 50 amino acids, but there are some that contain thousands. The absence, addition, or replacement of a single amino acid in the structure of a protein causes the protein to become a useless molecular heap. Every amino acid has to be in the right place and in the right order. The theory of evolution, which claims that life emerged as a result of chance, is quite helpless in the face of this order, since it is too wondrous to be explained by coincidence. Professor Fred Hoyle comments as follows: Indeed, such a theory, that life was assembled by an intelligence, is so obvious that one wonders why it is not widely accepted as being self-evident. The reasons are psychological rather than scientific. 242 The reason Hoyle used the term psychological is the self-conditioning of evolutionists not to accept that life was created. The rejection of Allah's existence is their main goal. For this reason alone, they go on defending irrational theories which they at the same time acknowledge to be impossible. Left-handed proteins. Let us now examine in detail why the evolutionist scenario regarding the formation of proteins is impossible. Even the correct sequence of the right amino acids is still not enough for the formation of a functional protein molecule. In addition to these requirements, each of the 20 different types of amino acids present in the composition of proteins must be left-handed. There are two different types of amino acids as of all organic molecules called left-handed and right-handed. The difference between them is the mirror symmetry between their three-dimensional structures, which is similar to that of a person's right and left hands. Amino acids of either of those two types can easily bond with one another. But one astonishing fact that has been revealed by research is that all the proteins in plants and animals on this planet, from the simplest organism to the most complex, are made up of left-handed amino acids. If even a single right-handed amino acid gets attached to the structure of a protein, the protein is rendered useless. In a series of experiments, surprisingly, bacteria that were exposed to right-handed amino acids immediately destroyed them. In some cases, they produced usable left-handed amino acids from the fractured components. Let us for an instant suppose that life came about by chance as evolutionists claim it did. In this case, the right and left-handed amino acids that were generated by chance should be present in roughly equal proportions in nature. Therefore, all living things should have both right and left-handed amino acids in their constitution, because chemically it is possible for amino acids of both types to combine with each other. However, as we know, in the real world the proteins existing in all living organisms are made up only of left-handed amino acids. The question of how proteins can pick out only the left-handed ones from among all amino acids, and how not even a single right-handed amino acid gets involved in the life process, is a problem that still baffles evolutionists. Such a specific and conscious selection constitutes one of the greatest impasses facing the theory of evolution. Moreover, this characteristic of proteins makes the problem facing evolutionists with respect to chance even worse. In order for a meaningful protein to be generated, it is not enough for the amino acids to be present in a particular number and sequence, and to be combined together in the right three-dimensional design. Additionally, all these amino acids have to be left-handed, not even one of them can be right-handed. Yet there is no natural selection mechanism which can identify that a right-handed amino acid has been added to the sequence and recognize that it must therefore be removed from the chain. This situation once more eliminates for good the possibility of coincidence and chance. The Britannica Science Encyclopedia, which is an outspoken defender of evolution, states that the amino acids of all living organisms on Earth, and the building blocks of complex polymers such as proteins, have the same left-handed asymmetry. It adds that this is tantamount to tossing a coin a million times and always getting heads. The same encyclopedia states that it is impossible to understand why molecules become left-handed or right-handed, and that this choice is fascinatingly related to the origin of life on Earth. 243 If a coin always turns up heads when tossed a million times, is it more logical to attribute that to chance, or else to accept that there is conscious intervention going on? The answer should be obvious. However, obvious though it may be, evolutionists still take refuge in coincidence, simply because they do not want to accept the existence of conscious intervention. A situation similar to the left-handedness of amino acids also exists with respect to nucleotides, the smallest units of the nucleic acids, DNA and RNA. In contrast to proteins, in which only left-handed amino acids are chosen, in the case of the nucleic acids, the preferred forms of their nucleotide components are always right-handed. This is another fact that can never be explained by chance. The indispensability of the peptide link, the difficulties the theory of evolution is unable to overcome with regard to the development of a single protein, are not limited to those we have recounted so far. It is not enough for amino acids to be arranged in the correct numbers, sequences, and required three-dimensional structures. The formation of a protein also requires that amino acid molecules with more than one arm be linked to each other only in certain ways. Such a bond is called a peptide bond. Amino acids can make different bonds with each other, but proteins are made up of those and only those amino acids which are joined by peptide bonds. A comparison will clarify this point. Suppose that all the parts of a car were complete and correctly assembled, with the sole exception that one of the wheels was fastened in place not with the usual nuts and bolts, but with a piece of wire, in such a way that its hub faced the ground. It would be impossible for such a car to move even the shortest distance, no matter how complex its technology or how powerful its engine. At first glance, everything would seem to be in the right place, but the faulty attachment of even one wheel would make the entire car useless. In the same way, in a protein molecule, the joining of even one amino acid to another with a bond other than a peptide bond would make the entire molecule useless. Research has shown that amino acids combining at random combine with the peptide bond only 50% of the time, and that the rest of the time different bonds that are not present in proteins emerge. To function properly, each amino acid making up a protein must be joined to others only with the peptide bond, in the same way that it likewise must be chosen only from among left-handed forms. A protein cannot form even if all the necessary conditions were present, since some people are unable to take a broad view of those matters, but approach them from a superficial viewpoint and assume protein formation to be a simple chemical reaction. They may make unrealistic deductions such as amino acids combined by way of reaction and then form proteins. However, accidental chemical reactions taking place in a non-living structure can only bring about simple compounds. The number of these is predetermined and limited. For a somewhat more complex chemical material, huge factories, chemical plants, and laboratories have to be involved. Medicines and many other chemical materials that we use in our daily life are made in just this way. Proteins have much more complex structures than these chemicals produced by industry. Therefore, it is impossible for proteins, each of which is a wonder of creation and engineering, in which every part takes its place in a fixed order, to originate as a result of haphazard chemical reactions. To summarize the subject of proteins, around 100 special proteins are needed for a single protein to form. Protein cannot form if even one of these enzymes, proteins, required for protein synthesis is missing. It is not enough for these 100 enzymes to be present at the same time, they must all also be present in a special region inside the cell, a specific region inside the nucleus. DNA manufactures the enzymes necessary for protein to form. Proteins are also needed for DNA replication. There is no possibility of one appearing before the other. Both have to be present at the same time. A ribosome that serves as a factory for protein formation must also exist. But the ribosome is itself made up of proteins. Therefore, proteins are needed for ribosomes to exist, and ribosomes are needed for proteins. It is impossible for one to form before the other. Proteins, DNA, the ribosome, the cell nucleus, mitochondria that produce energy and all the other organelles in the cell must all exist at one and the same time. The enzymes essential for protein to form have to be sent to the region where manufacture will be carried out by the cell. Even if enzymes are present, so long as they are not given tasks to perform by the cell, they will do nothing for that protein. There have to be a specific temperature and pH value in order for enzymes to be able to carry out reactions. Enzymes do not initiate reactions if they are not at the right temperature and pH level. Therefore, it is impossible for a protein to emerge so long as all the organelles of the cell do not coexist together. Even if we place all the components necessary for protein in some muddy water, these components can never combine together to constitute proteins. The existence of the cell is a prerequisite for that to happen. Amino acids do not normally react with one another. Helper enzymes to carry out a reaction have to be ready and present inside the cell. But they do naturally enter into reactions with various substances, such as sugar. Therefore, even if all the requisite amino acids are placed into muddy water, they can still never combine spontaneously with other amino acids. The cell is again essential for that to happen. Under natural conditions, even if a protein is left inside muddy water, that protein will immediately be broken down, under the effect of various environmental factors, or else will combine with other acids, amino acids or chemical substances and lose all its properties and turn into another substance that serves no purpose. In addition to all this, it will be useful to reiterate the essential conditions for a protein. A. There must be peptide bonds between amino acids. B. All amino acids must be left-handed. C. Only 20 amino acids must be used. D. Amino acids have to be in a specific sequence. C. The protein that forms has to have a specific 3D shape. Let us for a minute put aside all the impossibilities we have described so far, and suppose that a useful protein molecule still evolved spontaneously by accident. Even so, the theory of evolution again has no answers, because in order for this protein to survive, it would need to be isolated from its natural habitat and be protected under very special conditions. Otherwise, it would either disintegrate from exposure to natural conditions on Earth, or else join with other acids, amino acids, or chemical compounds, thereby losing its particular properties and turning into a totally different and useless substance. What we have been discussing so far is the impossibility of just one proteins coming about by chance. However, in the human body alone there are some 100,000 proteins functioning. Furthermore, there are about 1.5 million species named, and another 10 million are believed to exist. Although many similar proteins are used in many life forms, it is estimated that there must be 100 million or more types of protein in the plant and animal worlds. And the millions of species which have already become extinct are not included in this calculation. In other words, hundreds of millions of protein codes have existed in the world. If one considers that not even one protein can be explained by chance, it is clear what the existence of hundreds of millions of different proteins must mean. Bearing this truth in mind, it can clearly be understood that coincidences cannot account for the origin of living things. The evolutionary argument about the origin of life. Above all, there is one important point to take into consideration. If any one step in the evolutionary process is proven to be impossible, this is sufficient to prove that the whole theory is totally false and invalid. For instance, by proving that the haphazard formation of proteins is impossible, all other claims regarding the subsequent steps of evolution are also refuted. After this, it becomes meaningless to take some human and ape skulls and engage in speculation about them. How living organisms came into existence out of non-living matter was an issue that evolutionists did not even want to mention for a long time. However, this question, which had constantly been avoided, eventually had to be addressed, and attempts were made to settle it with a series of experiments in the second quarter of the 20th century. The main question was, how could the first living cell have appeared in the primordial atmosphere on the Earth? In other words, what kind of explanation could evolutionists offer? The first person to take the matter in hand was the Russian biologist Alexander I. Oparin, the founder of the concept of chemical evolution. Despite all his theoretical studies, Oparin was unable to produce any results to shed light on the origin of life. He says the following in his book The Origin of Life, published in 1936, "Unfortunately, however, the problem of the origin of the cell is perhaps the most obscure point in the whole study of the evolution of organisms." 244 Since Oparin, evolutionists have performed countless experiments, conducted research, and made observations to prove that a cell could have been formed by chance. However, every such attempt only made the complex structure of the cell clearer, and thus refuted the evolutionists' hypotheses even more. Professor Klaus Does, the president of the Institute of Biochemistry at the University of Johannes Gutenberg, states, more than 30 years of experimentation on the origin of life in the fields of chemical and molecular evolution have led to a better perception of the immensity of the problem of the origin of life on Earth rather than to its solution. At present, all discussions on principal theories and experiments in the field either end in stalemate or in a confession of ignorance. 245 In his book The End of Science, the evolutionary science writer John Horgan says of the origin of life, "This is by far the weakest strut of the chassis of modern biology." 246 The following statement by the geochemist Jeffrey Botta, from the San Diego-based Scripps Institute, makes the helplessness of evolutionists clear. Today, as we leave the 20th century, we still face the biggest unsolved problem that we had when we entered the 20th century. How did life originate on Earth? 247 Let us now look at the details of the theory of evolution's biggest unsolved problem. The first subject we have to consider is the famous Miller experiment. Miller's experiment, the most generally respected study on the origin of life, is the Miller experiment conducted by the American researcher Stanley Miller in 1953. The experiment is also known as the Urey-Miller experiment because of the contribution of Miller's instructor at the University of Chicago, Harold Urey. This experiment is the only evidence evolutionists have with which to allegedly prove the chemical evolution thesis. They advance it as the first stage of the supposed evolutionary process leading to life. Although nearly half a century has passed, and great technological advances have been made, nobody has made any further progress. In spite of this, Miller's experiment is still taught in textbooks as the evolutionary explanation of the earliest generation of living things. That is because, aware of the fact that such studies do not support, but rather actually refute, their thesis, evolutionist researchers deliberately avoid embarking on such experiments. Stanley Miller's aim was to demonstrate by means of an experiment that amino acids, the building blocks of proteins, could have come into existence by chance on the lifeless Earth billions of years ago. In his experiment, Miller used a gas mixture that he assumed to have existed on the primordial Earth, but which later proved unrealistic, composed of ammonia, methane, hydrogen, and water vapor. Since these gases would not react with each other under natural conditions, he added energy to the mixture to start a reaction among them. Supposing that this energy could have come from lightning in the primordial atmosphere, he used an electric current for this purpose. Miller heated this gas mixture at 100 degrees Celsius for a week and added the electrical current. At the end of the week, Miller analyzed the chemicals which had formed at the bottom of the jar, and observed that 3 out of the 20 amino acids which constitute the basic elements of proteins had been synthesized. This experiment aroused great excitement among evolutionists, and was promoted as an outstanding success. Moreover, in a state of intoxicated euphoria, various publications carried headlines such as "Miller creates life." However, what Miller had managed to synthesize was only a few inanimate molecules. Encouraged by this experiment, evolutionists immediately produced new scenarios. Stages following the development of amino acids were hurriedly hypothesized. Supposedly, amino acids had later united in the correct sequences by accident to form proteins. Some of these proteins which emerged by chance formed themselves into cell membrane-like structures which somehow came into existence and formed a primitive cell. These cells then supposedly came together over time to form multicellular living organisms. However, Miller's experiment has since proven to be false in many respects. For facts that invalidate Miller's experiment sought to prove that amino acids could form on their own in primordial Earth-like conditions, but it contains inconsistencies in a number of areas. 1. By using a mechanism called a cold trap, Miller isolated the amino acids from the environment as soon as they were formed. Had he not done so, the conditions in the environment in which the amino acids were formed would immediately have destroyed these molecules. Doubtless, this kind of conscious isolation mechanism did not exist on the primordial Earth. Without such a mechanism, even if one amino acid were obtained, it would immediately have been destroyed. The chemist Richard Bliss expresses this contradiction by observing that actually, without this trap, the chemical products would have been destroyed by the energy source. 248 And, sure enough, in his previous experiments, Miller had been unable to make even one single amino acid using the same materials without the cold trap mechanism. 2. The primordial atmosphere that Miller attempted to simulate in his experiment was not realistic. In the 1980s, scientists agreed that nitrogen and carbon dioxide should have been used in this artificial environment instead of methane and ammonia. So why did Miller insist on these gases? The answer is simple: without ammonia, it was impossible to synthesize any amino acid. Kevin McKean talks about this in an article published in Discover magazine. Miller and Urey imitated the ancient atmosphere on the Earth with a mixture of methane and ammonia. However, in the latest studies, it has been understood that the Earth was very hot at those times, and that it was composed of melted nickel and iron. Therefore, the chemical atmosphere of that time should have been formed mostly of nitrogen, N2, carbon dioxide, CO2, and water vapor, H2O. However, these are not as appropriate as methane and ammonia for the production of organic molecules. 249 The American scientists J.P. Ferris and C.T. Chen repeated Miller's experiment with an atmospheric environment that contained carbon dioxide, hydrogen, nitrogen, and water vapor, and were unable to obtain even a single amino acid molecule. 253 Another important point that invalidates Miller's experiment is that there was enough oxygen to destroy all the amino acids in the atmosphere at the time when they were thought to have been formed. This fact, overlooked by Miller, is revealed by the traces of oxidized iron found in rocks that are estimated to be 3.5 billion years old. 251 There are other findings showing that the amount of oxygen in the atmosphere at that time was much higher than originally claimed by evolutionists. Studies also show that the amount of ultraviolet radiation to which the Earth was then exposed was 10,000 times more than evolutionists' estimates. This intense radiation would unavoidably have freed oxygen by decomposing the water vapor and carbon dioxide in the atmosphere. This situation completely negates Miller's experiment, in which oxygen was completely neglected. If oxygen had been used in the experiment, methane would have decomposed into carbon dioxide and water, and ammonia into nitrogen and water. On the other hand, in an environment where there was no oxygen, there would be no ozone layer either. Therefore, the amino acids would have immediately been destroyed, since they would have been exposed to the most intense ultraviolet rays without the protection of the ozone layer. In other words, with or without oxygen in the primordial world, the result would have been a deadly environment for the amino acids. For at the end of Miller's experiment, many organic acids had also been formed with characteristics detrimental to the structure and function of living things. If the amino acids had not been isolated, and had been left in the same environment with these chemicals, their destruction or transformation into different compounds through chemical reactions would have been unavoidable. Moreover, Miller's experiment also produced right-handed amino acids. 252 The existence of those amino acids refuted the theory even within its own terms, because right-handed amino acids cannot function in the composition of living organisms. To conclude, the circumstances in which amino acids were formed in Miller's experiment were not suitable for life. In truth, this medium took the form of an acidic mixture, destroying and oxidizing the useful molecules obtained. All these facts point to one firm truth: Miller's experiment cannot claim to have proved that living things formed by chance under primordial Earth-like conditions. The whole experiment is nothing more than a deliberate and controlled laboratory experiment to synthesize amino acids. The amount and types of the gases used in the experiment were ideally determined to allow amino acids to originate. The amount of energy supplied to the system was neither too much nor too little, but arranged precisely to enable the necessary reactions to occur. The experimental apparatus was isolated, so that it would not allow the leaking of any harmful, destructive, or any other kind of elements to hinder the formation of amino acids. No elements, minerals or compounds that were likely to have been present on the primordial Earth, but which would have changed the course of the reactions, were included in the experiment. Oxygen, which would have prevented the formation of amino acids because of oxidation, is only one of these destructive elements. Even under such ideal laboratory conditions, it was impossible for the amino acids produced to survive and avoid destruction without the cold trap mechanism. In fact, by his experiment, Miller destroyed evolution's claim that life emerged as a result of unconscious coincidences. That is because, if the experiment proves anything, it is that amino acids can only be produced in a controlled laboratory environment where all the conditions are specifically designed by conscious intervention. Today, Miller's experiment is totally disregarded even by evolutionist scientists. In the February 1998 issue of the famous evolutionist science journal Earth, the following statements appear in an article titled Life's Crucible. Geologists now think that the primordial atmosphere consisted mainly of carbon dioxide and nitrogen, gases that are less reactive than those used in the 1953 experiment. And even if Miller's atmosphere could have existed, how do you get simple molecules such as amino acids to go through the necessary chemical changes that will convert them into more complicated compounds, or polymers, such as proteins? Miller himself throws up his hands at that part of the puzzle. It's a problem, he sighs with exasperation. How do you make polymers? That's not so easy. 253 As seen, today even Miller himself has accepted that his experiment does not lead to an explanation of the origin of life. In the March 1998 issue of National Geographic, in an article titled The Emergence of Life on Earth, the following comments appear: many scientists now suspect that the early atmosphere was different to what Miller first supposed. They think it consisted of carbon dioxide and nitrogen rather than hydrogen, methane, and ammonia. That's bad news for chemists. When they try sparking carbon dioxide and nitrogen, they get a paltry amount of organic molecules, the equivalent of dissolving a drop of food coloring in a swimming pool of water. Scientists find it hard to imagine life emerging from such a diluted soup. 254 In brief, neither Miller's experiment, nor any other similar one that has been attempted, can answer the question of how life emerged on Earth. All of the research that has been done shows that it is impossible for life to emerge by chance, and thus confirms that life is created. The reason evolutionists do not accept this obvious reality is their blind adherence to prejudices that are totally unscientific. Interestingly enough, Harun Yahya, who organized the Miller experiment with his student Stanley Miller, made the following confession on this subject: all of us who study the origin of life find that the more we look into it, the more we feel it is too complex to have evolved anywhere. We all believe as an article of faith that life evolved from dead matter on this planet. It is just that its complexity is so great, it is hard for us to imagine that it did. 255 The primordial atmosphere and proteins. Evolutionist sources use the Miller experiment, despite all of its inconsistencies, to try to gloss over the question of the origin of amino acids. By giving the impression that the issue has long since been resolved by that invalid experiment, they try to paper over the cracks in the theory of evolution. However, to explain the second stage of the origin of life, evolutionists faced an even greater problem than that of the formation of amino acids, namely, the origin of proteins, the building blocks of life, which are composed of hundreds of different amino acids bonding with each other in a particular order. Claiming that proteins were formed by chance under natural conditions is even more unrealistic and unreasonable than claiming that amino acids were formed by chance. In the preceding pages we have already seen the impossibility of the haphazard formation of proteins. Now, we will further examine the impossibility of proteins being produced chemically under primordial Earth conditions. The problem of protein synthesis in water. As we saw before, when combining to form proteins, amino acids form a special bond with one another called the peptide bond. A water molecule is released during the formation of this peptide bond. This fact definitely refutes the evolutionist explanation that primordial life originated in water, because, according to the Le Chatelier principle in chemistry, it is not possible for a reaction that releases water, a condensation reaction, to take place in a hydrous environment. The probability of this kind of a reaction happening in a hydrate environment is said to have the least probability of occurring of all chemical reactions. Hence the ocean, which is claimed to be where life began and amino acids originated, is definitely not an appropriate setting for amino acids to form proteins. 256 On the other hand, it would be irrational for evolutionists to change their minds and claim that life originated on land, because the only environment where amino acids could have been protected from ultraviolet radiation is in the oceans and seas. On land, they would be destroyed by ultraviolet rays. The Le Chatelier principle, on the other hand, disproves the claim of the formation of life in the sea. This is another dilemma confronting evolution. Fox's experiment challenged by the above-mentioned dilemma. Evolutionists began to invent unrealistic scenarios based on this water problem that so definitively refuted their theories. Sidney Fox was one of the best known of these researchers. Fox advanced the following theory to solve the problem. According to him, the first amino acids must have been transported to some cliffs near a volcano right after their formation in the primordial ocean. The water contained in this mixture that included the amino acids must have evaporated when the temperature increased above boiling point on the cliffs. The amino acids, which were dried out in this way, could then have combined to form proteins. However, this complicated way out was not accepted by many people in the field, because the amino acids could not have endured such high temperatures. Research confirmed that amino acids are immediately destroyed at very high temperatures. But Fox did not give up. He combined purified amino acids in the laboratory, under very special conditions, by heating them in a dry environment. The amino acids combined, but still no proteins were obtained. What he actually ended up with was simple and disordered loops of amino acids, arbitrarily combined with each other, and these loops were far from resembling any living protein. Furthermore, if Fox had kept the amino acids at a steady temperature, then these useless loops would also have disintegrated. Another point that nullified the experiment was that Fox did not use the useless end products obtained in Miller's experiment, Rather, he used pure amino acids from living organisms. This experiment, however, which was intended to be a continuation of Miller's experiment, should have started out from the results obtained by Miller. Yet neither Fox, nor any other researcher, used the useless amino acids Miller produced. Fox's experiment was not even welcomed in evolutionist circles, because it was clear that the meaningless amino acid chains that he obtained, which he termed proteinoids, could not have formed under natural conditions. Moreover, proteins, the basic units of life, still could not be produced. The problem of the origin of proteins remained unsolved. In an article in the popular science magazine, Chemical Engineering News, which appeared in the 1970s, Fox's experiment was mentioned as follows. Sidney Fox and the other researchers managed to unite the amino acids in the shape of proteinoids by using very special heating techniques under conditions which in fact did not exist at all in the primordial stages of Earth. Also, they are not at all similar to the very regular proteins present in living things. They are nothing but useless, irregular chemical stains. It was explained that even if such molecules had formed in the early ages, they would definitely be destroyed. 257 Indeed, the proteinoids Fox obtained were totally different from real proteins, both in structure and function. The difference between proteins and these proteinoids was as huge as the difference between a piece of high-tech equipment and a heap of unprocessed iron. Furthermore, it was impossible for these irregular amino acid chains to have survived in the primordial atmosphere. Harmful and destructive physical and chemical effects caused by heavy exposure to ultraviolet light and other unstable natural conditions would have caused these proteinoids to disintegrate. Because of the Vachatlier principle, it was also impossible for the amino acids to combine underwater, where ultraviolet rays would not reach them. In view of this, the idea that the proteinoids were the basis of life eventually lost support among scientists. The origin of the DNA molecular examinations so far have shown that the theory of evolution is in a serious quandary at the molecular level. Evolutionists have shed no light on the formation of amino acids at all. The formation of proteins, on the other hand, is another mystery all its own. Yet the problems are not even limited just to amino acids and proteins, these are only the beginning. Beyond them, the extremely complex structure of the cell leads evolutionists to yet another impasse. The reason for this is that the cell is not just a heap of amino acid-structured proteins, but rather one of the most complex systems man has ever encountered. While the theory of evolution was having such trouble providing a coherent explanation for the existence of the molecules that are the basis of the cell structure, developments in the science of genetics and the discovery of nucleic acids, DNA and RNA, produced brand new problems for the theory. In 1953, James Watson and Francis Crick launched a new age in biology with their work on the structure of DNA. The molecule known as DNA, which is found in the nucleus of each of the 100 trillion cells in our bodies, contains the complete blueprint for the construction of the human body. The information regarding all the characteristics of a person, from physical appearance to the structure of the inner organs, is recorded in DNA within the sequence of 4 special bases that make up the giant molecule. These bases are known as A, T, G, and C, according to the initial letters of their names. All the structural differences among people depend on variations in the sequences of these letters. In addition to features such as height, and eye, hair and skin colors, the DNA in a single cell also contains the design of the 206 bones, the 600 muscles, the 100 billion nerve cells, neurons, 1.000 trillion connections between the neurons of the brain, 97,000 kilometers of veins, and the 100 trillion cells of the human body. If we were to write down the information coded in DNA, then we would have to compile a giant library consisting of 900 volumes of 500 pages each. But the information this enormous library would hold is encoded inside the DNA molecules in the cell nucleus, which is far smaller than the 1/100th of a millimeter long cell itself. DNA cannot be explained by coincidences at this point, there is an important detail that deserves attention. An error in the sequence of the nucleotides making up a gene would render that gene completely useless. When it is considered that there are some 30,000 genes in the human body, it becomes clearer how impossible it is for the millions of nucleotides making up these genes to have been formed, in the right sequence, by chance. The impossibility of the formation of RNA and DNA by a coincidental accumulation of nucleotides is expressed by the French scientist Paul Auger in this way: we have to sharply distinguish the two stages in the chance formation of complex molecules such as nucleotides by chemical events. The production of nucleotides one by one, which is possible in the combination of these within very special sequences. The second is absolutely impossible. 258 For many years, Francis Crick believed in the theory of molecular evolution, but eventually even he had to admit to himself that such a complex molecule could not have emerged spontaneously by chance, as the result of an evolutionary process. An honest man, armed with all the knowledge available to us now, could only state that, in some sense, the origin of life appears at the moment to be almost a miracle. 259 The Turkish evolutionist Professor Ali Demirsoy was forced to make the following confession on the issue: in fact, the probability of the formation of a protein and a nucleic acid, DNA-RNA, is a probability way beyond estimating. Furthermore, the probability of the emergence of a certain protein chain is so slight as to be called astronomic. 260 A very interesting paradox emerges at this point: while DNA can only replicate with the help of special proteins, enzymes, the synthesis of these proteins can only be realized by the information encoded in DNA. As they both depend on each other, they have to exist at the same time for replication. Science writer John Horgan explains the dilemma in this way: DNA cannot do its work, including forming more DNA, without the help of catalytic proteins, or enzymes. In short, proteins cannot form without DNA, but neither can DNA form without proteins. 261 This situation once again undermines the scenario that life could have come about by accident. Homer Jacobson, Professor Emeritus of Chemistry, comments, "Directions for the reproduction of plants, for energy and the extraction of parts from the current environment, for the growth sequence, and for the effector mechanism translating instructions into growth all had to be simultaneously present at that moment, when life began. This combination of events has seemed an incredibly unlikely happenstance. 262 The quotation above was written 2 years after the discovery of the structure of DNA by Watson and Crick. But despite all the developments in science, this problem for evolutionists remains unsolved. This is why German biochemist Douglas R. Hofstadter says, "How did the genetic code, along with the mechanisms for its translation, ribosomes and RNA molecules, originate?" For the moment, we will have to content ourselves with the sense of wonder and awe, rather than with an answer. 263 Stanley Miller and Francis Crick's close associate from the University of San Diego, California, the highly reputed evolutionist Dr. Leslie Orgel says in an article published in 1994, "It is extremely improbable that proteins and nucleic acids, both of which are structurally complex, arose spontaneously in the same place at the same time. Yet it also seems impossible to have one without the other. And so, at first glance, one might have to conclude that life could never, in fact, have originated by chemical means." 264 Alongside all of this, it is chemically impossible for nucleic acids such as DNA and RNA, which possess a definite string of information, to have emerged by chance, or for even one of the nucleotides which compose them to have come about by accident and to have survived and maintained its unadulterated state under the conditions of the primordial world. Even the famous journal Scientific American, which follows an evolutionist line, has been obliged to confess the doubts of evolutionists on this subject. Even the simpler molecules are produced only in small amounts in realistic experiments simulating possible primitive Earth conditions. What is worse, these molecules are generally minor constituents of TARS, It remains problematical how they could have been separated and purified through geochemical processes whose normal effects are to make organic mixtures more and more of a jumble. With somewhat more complex molecules, these difficulties rapidly increase. In particular, a purely geochemical origin of nucleotides, the subunits of DNA and RNA, presents great difficulties. 265 As revealed by what has been discussed so far, since it is impossible for life to have emerged by chemical means, life was created by all-powerful Allah. This chemical evolution that evolutionists have been talking about since the beginning of the last century never happened, and is nothing but a myth. But most evolutionists believe in this and similar totally unscientific fairy tales as if they were true, because accepting that living things were created means accepting Almighty Allah's existence, and they have conditioned themselves not to accept this truth. One famous biologist from Australia, Michael Denton, discusses the subject in his book Evolution, A Theory in Crisis. To the skeptic, the proposition that the genetic programs of higher organisms, consisting of something close to 1,000 million bits of information, equivalent to the sequence of letters in a small library of 1,000 volumes, containing in encoded form countless thousands of intricate algorithms controlling, specifying, and ordering the growth and development of billions and billions of cells into the form of a complex organism, were composed by a purely random process is simply an affront to reason. But to the Darwinist, the idea is accepted without a ripple of doubt. The paradigm takes precedence! 266 The invalidity of the RNA world. The discovery in the 1970s that the gases originally existing in the primitive atmosphere of the Earth would have rendered amino acid synthesis impossible was a serious blow to the theory of molecular evolution. Evolutionists then had to face the fact that the primitive atmosphere experiments by Stanley Miller, Sidney Fox, Cyril Ponampuruma and others were invalid. For this reason, in the 1980s the evolutionists tried again. As a result, the RNA world hypothesis was advanced. This scenario proposed that, not proteins, but rather the RNA molecules that contain the information for proteins, were formed first. According to this scenario, advanced by Harvard chemist Walter Gilbert in 1986, inspired by the discovery about ribozymes by Thomas Seck, billions of years ago an RNA molecule capable of replicating itself formed somehow by accident. Then this RNA molecule started to produce proteins, having been activated by external influences. Thereafter, it became necessary to store this information in a second molecule, and somehow the DNA molecule emerged to do that. Made up as it is of a chain of impossibilities in each and every stage, this scarcely credible scenario, far from providing any explanation of the origin of life, only magnified the problem, and raised many unanswerable questions. 1. Since it is impossible to accept the coincidental formation of even one of the nucleotides making up RNA, how can it be possible for these imaginary nucleotides to form RNA by coming together in a particular sequence? Evolutionist John Horgan admits the impossibility of the chance formation of RNA. As researchers continue to examine the RNA world concept closely, more problems emerge. How did RNA initially arise? RNA and its components are difficult to synthesize in a laboratory under the best of conditions, much less under really plausible ones. 267 Even if we suppose that it formed by chance, how could this RNA, consisting of just a nucleotide chain, have decided to self-replicate? And with what kind of mechanism could it have carried out this self-replicating process? Where did it find the nucleotides it used while self-replicating? Even evolutionist microbiologists Gerald Joyce and Leslie Orgel expressed the desperate nature of the situation in their book In the RNA World. This discussion has, in a sense, focused on a straw man, the myth of a self-replicating RNA molecule that arose de novo from a soup of random polynucleotides. Not only is such a notion unrealistic in light of our current understanding of prebiotic chemistry, but it would strain the credulity of even an optimist's view of RNA's catalytic potential. 268 Even if we suppose that there was self-replicating RNA in the primordial world, that numerous amino acids of every type ready to be used by RNA were available, and that all of these impossibilities somehow took place, the situation still does not lead to the formation of even one single protein. For RNA only includes information concerning the structure of proteins. Amino acids, on the other hand, are raw materials. Nevertheless, there is no mechanism for the production of proteins. To consider the existence of RNA sufficient for protein production is as nonsensical as expecting a car to assemble itself by simply throwing the blueprint onto a heap of parts piled up on top of each other. A blueprint cannot produce a car all by itself without a factory and workers to assemble the parts according to the instructions contained in the blueprint. In the same way, the blueprint contained in RNA cannot produce proteins by itself without the cooperation of other cellular components which follow the instructions contained in the RNA. Proteins are produced in the ribosome factory with the help of many enzymes, and as a result of extremely complex processes within the cell. The ribosome is a complex cell organelle made up of proteins. This leads, therefore, to another unreasonable supposition that ribosomes, too, should have come into existence by chance at the same time. Even Nobel Prize winner Jacques Monod, who was one of the most fanatical defenders of evolution and atheism, explained that protein synthesis can by no means be considered to depend merely on the information in the nucleic acids. The code is meaningless unless translated. The modern cell's translating machinery consists of at least 50 macromolecular components, which are themselves coded in DNA. The code cannot be translated otherwise than by products of translation themselves. It is the modern expression of Vami Vivum ex ovo. When and how did this circle become closed? It is exceedingly difficult to imagine. 269 How could an RNA chain in the primordial world have taken such a decision, and what methods could it have employed to make protein production happen by doing the work of 50 specialized particles on its own? Evolutionists have no answer to these questions. One article in the preeminent scientific journal Nature makes it clear that the concept of self-replicating RNA is a complete product of fantasy, and that actually this kind of RNA has not been produced in any experiment. DNA replication is so error-prone that it needs the prior existence of protein enzymes to improve the copying fidelity of a gene-sized piece of DNA. Catch 22. Say Maynard Smith and Sathbury. So, wheel on RNA with its now recognized properties of carrying both informational and enzymatic activity, leading the authors to state, in essence, the first RNA molecules did not need a protein polymerase to replicate them, they replicated themselves. Is this a fact or hope? I would have thought it relevant to point out for biologists in general that not one self-replicating RNA has emerged to date from quadrillions, 1024, of artificially synthesized, random RNA sequences. 270 Dr. Leslie Orgel uses the term scenario for the possibility of the origination of life through the RNA world. Orgel described what kind of features this RNA would have had to have and how impossible these would have been in his article The Origin of Life, published in Scientific American in October 1994. This scenario could have occurred, we noted, if prebiotic RNA had two properties not evident today: a capacity to replicate without the help of proteins and an ability to catalyze every step of protein synthesis. 271 As should by now be clear, to expect these two complex and extremely essential processes from a molecule such as RNA is against scientific thought. Concrete scientific facts, on the other hand, makes it explicit that the RNA world hypothesis, which is a new model proposed for the transformation of life, is an equally implausible fable. Job Horgan, in his book The End of Science, reports that Stanley Miller viewed the theories subsequently put forward regarding the origin of life as quite meaningless. It will be recalled that Miller was the originator of the famous Miller experiment, which was later revealed to be invalid. In fact, almost 40 years after his original experiment, Miller told me that solving the riddle of the origin of life had turned out to be more difficult than he or anyone else had envisioned. Miller seemed unimpressed with any of the current proposals on the origin of life, referring to them as nonsense or paper chemistry. He was so contemptuous of some hypotheses that, when I asked his opinion of them, he merely shook his head, sighed deeply, and snickered as if overcome by the folly of humanity. Stuart Kauffman's theory of autocatalysis fell into this category. Running equations through a computer does not constitute an experiment, Miller sniffed. Miller acknowledged that scientists may never know precisely where and when life emerged. 272 This statement, by a pioneer of the struggle to find an evolutionary explanation for the origin of life, clearly reflects the despair felt by evolutionist scientists over the cul-de-sac they find themselves in. Order cannot be explained by coincidence. So far, we have examined how impossible the accidental formation of life is. Let us again ignore these impossibilities for just a moment. Let us suppose that millions of years ago a cell was formed which had acquired everything necessary for life, and that it duly came to life. Evolution again collapses at this point. For even if this cell had existed for a while, it would eventually have died, and after its death, nothing would have remained, and everything would have reverted to where it had started. This is because this first living cell, lacking any genetic information, would not have been able to reproduce and start a new generation. Life would have ended with its death. The genetic system does not only consist of DNA. The following things must also exist in the same environment: enzymes to read the code on the DNA, messenger RNA to be produced after reading these codes, a ribosome to which messenger RNA will attach according to this code, transfer RNA to transfer the amino acids to the ribosome for use in production, and extremely complex enzymes to carry out numerous intermediary processes. Such an environment cannot exist anywhere apart from a totally isolated and completely controlled environment such as the cell, where all the essential raw materials and energy resources exist. As a result, organic matter can self-reproduce only if it exists as a fully developed cell, with all its organelles. This means that the first cell on Earth was formed all of a sudden, together with its extraordinarily complex structure. So, if a complex structure came into existence all of a sudden, what does this mean? Let us ask this question with an example. Let us liken the cell to a high-tech car in terms of its complexity. In fact, the cell is a much more complex and developed system than a car. Now let us ask the following question: what would you think if you went out hiking in the depths of a thick forest and ran across a brand new car among the trees? Would you imagine that various elements in the forest had come together by chance over millions of years and produced such a vehicle? All the parts in the car are made of products such as iron, copper, and rubber, the raw ingredients for which are all found on the Earth. But would this fact lead you to think that these materials had synthesized by chance and then come together and manufactured such a car? There is no doubt that anyone with a sound mind would realize that the car was the product of an intelligent design, and wonder what it was doing there in the middle of the forest. The sudden emergence of a complex structure in a complete form, quite out of the blue, shows that this is the work of an intelligent design. Believing that pure chance can produce perfect structures goes well beyond the bounds of reason. Yet every explanation put forward by the theory of evolution regarding the origin of life is like that. One outspoken authority on this issue is the famous French zoologist Pierre-Paul Grass. Grass is an evolutionist, yet he acknowledges that Darwinist theory is unable to explain life and makes a point about the logic of coincidence, which is the backbone of Darwinism. The opportune appearance of mutations permitting animals and plants to meet their needs seems hard to believe. Yet the Darwinian theory is even more demanding. A single plant, a single animal would require thousands and thousands of lucky, appropriate events. Thus, miracles would become the rule. Events with an infinitesimal probability could not fail to occur. There is no law against daydreaming, but science must not indulge in it. 273 All living things in the world, all of which are clear examples of the intelligent planning we have just been discussing, are at the same time living evidence that coincidence can have no role to play in their existence. Each of its component parts, never mind a whole living creature, contains structures and systems so complex that they cannot be the work of coincidence. We need go no further than our own bodies to find examples of this. One example of this is our eyes. The human eye sees by the working together of some 40 separate parts. If one of those is not present, the eye will be useless. Each of these 40 parts possesses complex structures within itself. The retina at the back of the eye, for instance, is made up of 11 layers. Each layer has a different function. The chemical processes that go on inside the retina are so complex that they can only be explained with pages full of formulae and diagrams. The theory of evolution is unable to account for the emergence of even such a flawless and complex structure as a single eye by means of accident, let alone life itself, or mankind. So, what do these extraordinary features in living things prove to us about the origin of life? As we made clear in the opening part of this book, only two different accounts can be given regarding the origin of life. One is the fallacious evolutionary explanation, the other the evident fact of creation. As explained throughout the book, the evolution claim is impossible, and scientific discoveries prove the truth of creation. This truth may surprise some scientists, who from the 19th century to the present have seen the concept of creation as unscientific, but science can only progress by overcoming shocks of this kind and accepting the truth. Chandra Wickramasinghe describes the reality he faced as a scientist who had been told throughout his life that life had emerged as a result of chance coincidences. From my earliest training as a scientist, I was very strongly brainwashed to believe that science cannot be consistent with any kind of deliberate creation. That notion has had to be painfully shed. At the moment, I can't find any rational argument to knock down the view which argues for conversion to Allah. We used to have an open mind, now we realize that the only logical answer to life is creation, and not accidental random shuffling. Chapter 9. The Myth of Homology. Anyone who studies the different living species in the world may observe that there are some similar organs and features among these species. The first person to draw materialistic conclusions from this fact, which has attracted scientists' attention since the 18th century, was Charles Darwin. Darwin thought that creatures with similar, homologous, organs had an evolutionary relationship with each other, and that these organs must have been inherited from a common ancestor. According to his assumption, both pigeons and eagles had wings, therefore, pigeons, eagles and indeed all other birds with wings were supposed to have evolved from a common ancestor. Homology is a tautological argument, advanced on the basis of no other evidence than an apparent physical resemblance. This argument has never once been verified by a single concrete discovery in all the years since Darwin's day. Nowhere in the world has anyone come up with the fossil remain of the imaginary common ancestor of creatures with homologous structures. Furthermore, the following issues make it clear that homology provides no evidence that evolution ever occurred. 1. One finds homologous organs in creatures belonging to completely different phyla, among which evolutionists have not been able to establish any sort of evolutionary relationship. 2. The genetic codes of some creatures that have homologous organs are completely different from one another. 3. The embryological development of homologous organs in different creatures is completely different. Let us now examine each of these points one by one. The invalidity of morphological homology. The homology thesis of the evolutionists is based on the logic of building an evolutionary link between all living things with similar morphologies, structures, whereas there are a number of homologous organs shared by different groups that are completely unrelated to each other. Wings are one example. In addition to birds, we find wings on bats, which are mammals, and on insects and even on some dinosaurs, which are extinct reptiles. Not even evolutionists posit an evolutionary relationship or kinship among those four different groups of animals. Another striking example is the amazing resemblance and the structural similarity observed in the eyes of different creatures. For example, the octopus and man are two extremely different species, between which no evolutionary relationship is likely even to be proposed. yet the eyes of both are very much alike in terms of their structure and function. Not even evolutionists try to account for the similarity of the eyes of the octopus and man by positing a common ancestor. In response, evolutionists say that these organs are not homologous, in other words, from a common ancestor, but that they are analogous, very similar to each other, although there is no evolutionary connection between them. For example, in their view, the human eye and the octopus eye are analogous organs. However, the question of which category they will put an organ into, homologous or analogous, is answered totally in line with the theory of evolution's preconceptions. And this shows that the evolutionist claim based on resemblances is completely unscientific. The only thing evolutionists do is to try to interpret new discoveries in accordance with a dogmatic evolutionary preconception. However, the interpretation they put forward is completely invalid, because organs which they have to consider analogous sometimes bear such close resemblance to one another, despite being exceedingly complex structures, that it is totally illogical to propose that this similarity was brought about as a result of coincidental mutations. If an octopus eye emerged completely by coincidence, as evolutionists claim, then how is it that vertebrate eyes can emerge by the very same coincidences? The famous evolutionist Frank Salisbury, who got dizzy from thinking about this question, writes, "Even something as complex as the eye has appeared several times, for example, in the squid, the vertebrates, and the arthropods. It's bad enough accounting for the origin of such things once, but the thought of producing them several times according to the modern synthetic theory makes my head swim." 275 According to the theory of evolution, wings emerged independently of each other 4 times, in insects, flying reptiles, birds, and flying mammals, bats. The fact that wings with very similar structures developed 4 times which cannot be explained by the mechanisms of natural selection, mutation is yet another headache for evolutionary biologists. One of the most concrete examples of such an obstacle in the path of evolutionary theory can be seen in mammals. According to the accepted view of modern biology, all mammals belong to one of 3 basic categories: placentals, marsupials and monotremes. Evolutionists consider this distinction to have come about when mammals first appeared, and that each group lived its own evolutionary history totally independent of the other. But it is interesting that there are pairs in placentals and marsupials which are nearly the same. Placental wolves, cats, squirrels, anteaters, moles and mice all have their marsupial counterparts with closely similar morphologies. 276 In other words, according to the theory of evolution, mutations completely independent of each other must have produced these creatures by chance twice. This reality is a question that will give evolutionists problems even worse than dizzy spells. One of the interesting similarities between placental and marsupial mammals is that between the North American wolf and the Tasmanian wolf. The former belongs to the placental class, the latter to the marsupials. Evolutionary biologists believe that these 2 different species have completely separate evolutionary histories. 277 Since the continent of Australia and the islands around it split off from Gondwanaland, the supercontinent that is supposed to be the originator of Africa, Antarctica, Australia, and South America, the link between placental and marsupial mammals is considered to have been broken, and at that time there were no wolves. But the interesting thing is that the skeletal structure of the Tasmanian wolf is nearly identical to that of the North American wolf. Their skulls in particular, as shown on the next page, bear an extraordinary degree of resemblance to each other. Extraordinary resemblances and similar organs like these, which evolutionary biologists cannot accept as examples of homology, show that homology does not constitute any evidence for the thesis of evolution from a common ancestor. What is even more interesting is that the exact opposite situation is to be observed in other living things. In other words, there are living things, some of whose organs have completely different structures, even though they are considered to be close relatives by evolutionists. For example, most crustaceans have eye structures of the refracting lens type. In only 2 species of crustacean, the lobster and the shrimp, is the completely different reflecting type of eye seen. See the chapter on irreducible complexity, the genetic and embryological impasse of homology. The discovery which really overthrew homology is that organs accepted as homologous are almost all controlled by very different genetic codes. As we know, the theory of evolution proposes that living things develop through small, chance changes in their genes, in other words, mutations. For this reason, the genetic structures of living things which are seen as close evolutionary relatives should resemble each other. And, in particular, similar organs should be controlled by similar genetic structures. However, in point of fact, genetic researchers have made discoveries which conflict totally with this evolutionary thesis. Similar organs are usually governed by very different genetic, DNA, codes. Furthermore, similar genetic codes in the DNA of different creatures are often associated with completely different organs. The chapter titled The Failure of Homology in Michael Denton's book, Evolution, A Theory in Crisis, gives several examples of this, and sums the subject up in this way. Homologous structures are often specified by non-homologous genetic systems, and the concept of homology can seldom be extended back into embryology. 278 This genetic question has also been raised by the well-known evolutionary biologist Gavin Beer. In his book Homology, An Unsolved Problem, published in 1971, De Beer put forward a very wide-ranging analysis of this subject. He sums up why homology is a problem for the theory of evolution as follows: what mechanism can it be that results in the production of homologous organs, the same patterns, in spite of their not being controlled by the same genes? I asked this question in 1938, and it has not been answered. 279 Although some 30 years have passed since De Beer wrote those words, they have still received no answer. A third proof which undermines the homology claim is the question of embryological development, which we mentioned at the start. In order for the evolutionary thesis regarding homology to be taken seriously, the periods of similar structures embryological development, in other words, the stages of development in the egg or the mother's womb, would need to be parallel, whereas, in reality, these embryological periods for similar structures are quite different from each other in every living creature. Per Albert, an eminent developmental biologist, noted, it is the rule rather than the exception that homologous structures formed from distinctly dissimilar initial states. 280 The emergence of similar structures as a result of totally dissimilar processes is frequently seen in the latter stages of the development phase. As we know, many species of animal go through a stage known as indirect development, in other words the larva stage, on their way to adulthood. For instance, most frogs begin life as swimming tadpoles and turn into four-legged animals at the last stage of metamorphosis. But alongside this there are several species of frog which skip the larva stage and develop directly. But the adults of most of these species that develop directly are practically indistinguishable from those species which pass through the tadpole stage. The same phenomenon is to be seen in water chestnuts and some other similar species. 281 To conclude, we can say that genetic and embryological research has proven that the concept of homology defined by Darwin as evidence of the evolution of living things from a common ancestor can by no means be regarded as any evidence at all. The inconsistency of homology, which looks quite convincing on the surface, is clearly revealed when examined more closely. The fall of the homology in tetrapod limbs. We have already examined homology's morphological claim, in other words the invalidity of the evolutionist claim based on similarities of form and living things, but it will be useful to examine one well-known example of this subject a little more closely. This is the fore and hind limbs of the tetrapods, presented as a clear proof of homology in almost all books on evolution. Tetrapods, that is, land-living vertebrates, have 5 digits on their fore and hind limbs. Although these may not always look like fingers or toes, they are all counted as pentadactyl, 5-digit, due to their bone structure. The hands and feet of a frog, a lizard, a squirrel, or a monkey all have this same structure. Even the bone structures of birds and bats conform to this basic design. Evolutionists claim that all living things descended from a common ancestor, and they have long cited pentadactyl limb as evidence of this. But they know that this claim actually possesses no scientific validity. Even today, evolutionists accept the feature of pentadactylism in living things among which they have been able to establish no evolutionary link. For example, in two separate scientific papers published in 1991 and 1996, evolutionary biologist M. Coates reveals that pentadactylism emerged two separate times, each independently of the other. According to Coates, the pentadactyl structure emerged independently in anthroposaurs and amphibians. 282 This discovery is a sign that pentadactylism is no evidence for a common ancestor. Another matter which creates difficulties for the evolutionist thesis in this respect is that these creatures have 5 digits on both their fore and hind limbs. It is not proposed in evolutionist literature that fore and hind limb descended from a common limb. Rather, it is assumed that they developed separately. For this reason, it should be expected that the structure of the fore and hind limbs should be different, the result of different, chance mutations. Michael Denton has this to say on the subject: The forelimbs of all terrestrial vertebrates are constructed according to the same pentadactyl design, and this is attributed by evolutionary biologists as showing that all have been derived from a common ancestral source. But the hind limbs of all vertebrates also conform to the pentadactyl pattern and are strikingly similar to the forelimbs in bone structure and in their detailed embryological development. Yet no evolutionist claims that the hind limb evolved from the forelimb, or that hind limbs and forelimbs evolved from a common source. Invariably, as biological knowledge has grown, common genealogy as an explanation for similarity has tended to grow ever more tenuous. Like so much of the other circumstantial evidence for evolution, that drawn from homology is not convincing because it entails too many anomalies, too many counter-instances, far too many phenomena which simply do not fit easily into the orthodox picture. 283 But the real blow dealt to the evolutionist claim of the homology of pentadactylism came from molecular biology. The assumption of the homology of pentadactylism, which was long maintained in evolutionist publications, was overturned when it was realized that the limb structures were controlled by totally different genes in different creatures possessing this pentadactyl structure. Evolutionary biologist William Fix describes the collapse of the evolutionist thesis regarding pentadactylism in this way: the older textbooks on evolution make much of the idea of homology, pointing out the obvious resemblances between the skeletons of the limbs of different animals. Thus the pentadactyl, 5-bone, limb pattern is found in the arm of a man, the wing of a bird, and flipper of a whale, and this is held to indicate their common origin. Now if these various structures were transmitted by the same gene couples, varied from time to time by mutations and acted upon by environmental selection, the theory would make good sense. Unfortunately, this is not the case. Homologous organs are now known to be produced by totally different gene complexes in the different species. The concept of homology in terms of similar genes handed on from a common ancestor has broken down. 284 On closer examination, William Fix is saying that evolutionist claims regarding pentadactylism homology appeared in old textbooks, but that the claims were abandoned after molecular evidence emerged. But some evolutionist sources still continue to put it forward as major evidence for evolution. The invalidity of molecular homology. Evolutionists' advancement of homology as evidence for evolution is invalid not only at the morphological level, but also at the molecular level. Evolutionists say that the DNA codes, or the corresponding protein structures, of different living species are similar, and that this similarity is evidence that these living species have evolved from common ancestors, or else from each other. For example, it is regularly stated in the evolutionist literature that there is a great similarity between the DNA of a human and that of an ape, and this similarity is presented as a proof for the evolutionist claim that there is an evolutionary relationships between man and ape. We must make it clear from the start that it is perfectly natural that living creatures on the earth should possess very similar DNA structures. Living things' basic life processes are the same, and since human beings possess a living body, they cannot be expected to have a different DNA structure to other creatures. Like other creatures, human beings develop by consuming carbohydrates, lipids, and proteins. Oxygen circulates through the blood in their bodies, and energy is produced every second in each of their cells by the use of this oxygen. For this reason, the fact that living things possess genetic similarities is no proof of the evolutionist claim that they evolved from a common ancestor. If evolutionists want to prove their theory of evolution from a common ancestor, then they have to show that creatures alleged to be each other's common ancestors have a direct line of descent in their molecular structures. In fact, however, as we shall shortly be examining, there have been no concrete discoveries showing any such thing. Let us first of all take the matter of the similarity between human and chimpanzee DNA. The latest studies on this issue have revealed that evolutionist propaganda about a 98% or 99% similarity between man and chimp is totally erroneous. If a slightly wider study is made of this subject, it can be seen that the DNA of much more surprising creatures resembles that of man. One of these similarities is between man and worms of the nematode phylum. For example, genetic analyses published in New Scientist have revealed that nearly 75% of human genes have some counterpart in nematodes' millimeter-long soil-dwelling worms. 285. This definitely does not mean that there is only a 25% difference between man and these worms. According to the family tree made by evolutionists, the Chordata phylum, in which man is included, and the Nematoda phylum were different to each other even 530 million years ago. This situation clearly reveals that the similarity between the DNA strands of those two different categories of life is no evidence for the claim that these creatures evolved from a common ancestor. In fact, when the results of DNA analyses from different species and classes are compared, it is seen that the sequences clearly do not agree with any evolutionist family tree. According to the evolutionist thesis, living things must have undergone a progressive increase in complexity, and, parallel to this, it is to be expected that the number of genes, which make up their genetic data, should also gradually increase. But the data obtained show that this thesis is the work of fantasy. The Russian scientist Theodosius Dobzhansky, one of the best-known theoreticians of evolution, once stated that this irregular relationships between living things and their DNA is a great problem that evolution cannot explain. More complex organisms generally have more DNA per cell than do simpler ones, but this rule has conspicuous exceptions. Man is nowhere near the top of the list, being exceeded by Amphiuma, an amphibian, Protopterus, a lungfish, and even ordinary frogs and toads. Why this should be so has long been a puzzle. 286 Other comparisons on the molecular level produce other examples of inconsistency which render evolutionist views meaningless. When the protein strands of various living things are analyzed in a laboratory, results emerge which are totally unexpected from the evolutionist's point of view, and some of which are utterly astounding. For example, the cytochrome C protein in man differs by 14 amino acids from that in a horse, but by only 8 from that in a kangaroo. When the same strand is examined, turtles appear closer to man than to a reptile such as the rattlesnake. When this situation is viewed from the evolutionist point of view, a meaningless result will emerge, such as that turtles are more closely related to man than they are to snakes. For instance, chickens and sea snakes differ by 17 amino acids in 100 codons and horses and sharks by 16, which is a greater difference than that between dogs and worm flies, which belong to different phyla even, and which differ by only 15 amino acids. Similar facts have been discovered with respect to hemoglobin. The hemoglobin protein found in human beings differs from that found in lemurs by 20 amino acids, but from that in pigs by only 14. The situation is more or less the same for other proteins. 287 This being the case, evolutionists should arrive at the conclusion that, in evolutionary terms, man is more closely related to the kangaroo than to the horse, or to the pig than to the lemur. But these results conflict with all the evolutionary family tree plans that have so far been accepted. Protein similarities continue to produce astounding surprises. For example, Adrian Friday and Martin Bishop of Cambridge have analyzed the available protein sequence data for tetrapods. To their surprise, in nearly all cases, man, the mammal, and chicken, the bird, were paired off as closest relatives, with the crocodile as next nearest relative. 288 Again, when these similarities are approached from the point of view of evolutionist logic, they lead us to the ridiculous conclusion that man's closest evolutionary relative is the chicken. Paul Rich stresses the fact that molecular analyses produce results that show very different groups of living things to be closely related in this way. Proteins with nearly the same structure and function, homologous proteins, are found in increasing numbers in phylogenetically different, even very distinct taxa, for example, hemoglobins in vertebrates, in some invertebrates, and even in certain plants. 289 Dr. Christian Schwab, a biochemical researcher from the University of South Carolina's Faculty of Medicine, is a scientist who spent years trying to find evidence for evolution in the molecular field. He first tried to establish evolutionary relationships between living things by carrying out studies on proteins such as insulin and relaxin. But Schwab has several times been forced to admit that he has not been able to come by any evidence for evolution in his studies. He says the following in an article in Science: molecular evolution is about to be accepted as a method superior to paleontology for the discovery of evolutionary relationships. As a molecular evolutionist I should be elated. Instead it seems disconcerting that many exceptions exist to the orderly progression of species as determined by molecular homologies, so many in fact that I think the exception, the quirks, may carry the more important message. 290 Schwab's studies on relaxins produced rather interesting results. Against this background of high variability between relaxins from purportedly closely related species, the relaxins of pig and whale are all but identical. The molecules derived from rats, guinea pigs, man and pigs are as distant from each other, approximately 55%, as all are from the elasmo branch's relaxin. Insulin, however, brings man and pig phylogenetically closer together than chimpanzee and man. 291 Schwab was faced by the same realities when he compared the arrangements of other proteins besides insulin and relaxin. Schwab has this to say about these other proteins that constitute exceptions to the orderly molecular development proposed by evolutionists. The relaxin and insulin families do not stand alone as exceptions to the orderly interpretation of molecular evolution in conventional monophyletic terms. It is instructive to look at additional examples of purportedly anomalous protein evolution and note that the explanations permissible under the molecular clock theories cover a range of ad hoc explanations apparently limited only by imagination. 292 Schwab reveals that the comparison of the arrangement of lysosomes, cytochromes, and many hormones and amino acids show unexpected results and anomalies from the evolutionary point of view. Based on all this evidence, Schwab maintains that all proteins had their present forms right from the start, undergoing no evolution, and that no intermediate form has been found between molecules, in the same way as with fossils. Concerning these findings in the field of molecular biology, Dr. Michael Denton comments, each class at a molecular level is unique, isolated and unlinked by intermediates. Thus, molecules, like fossils, have failed to provide the elusive intermediates so long sought by evolutionary biology. At a molecular level, no organism is ancestral or primitive or advanced compared with its relatives. There is little doubt that if this molecular evidence had been available a century ago, the idea of organic evolution might never have been accepted. 293 The tree of life is collapsing. In the 1990s, research into the genetic codes of living things worsened the quandary faced by the theory of evolution in this regard. In these experiments, instead of the earlier comparisons that were limited to protein sequences, ribosomal RNA sequences were compared. From these findings, evolutionist scientists sought to establish an evolutionary tree. However, they were disappointed by the results. According to a 1999 article by French biologists Hervé Philippe and Patrick Fortre, With more and more sequences available, it turned out that most protein phylogenies contradict each other as well as the RNA tree. 294 Besides RNA comparisons, the DNA codes in the genes of living things were also compared, but the results have been the opposite of the tree of life presupposed by the theory of evolution. Molecular biologists James A. Lake, Ravi Jain and Maria C. Rivera elaborated on this in an article in 1999. As scientists started analyzing a variety of genes from different organisms and found that their relationship to each other contradicted the evolutionary tree of life derived from RNA analysis alone. 295 Neither the comparisons that have been made of proteins, nor those of RNAs or of genes, confirm the premises of the theory of evolution. Carl Moise, a highly reputed biologist from the University of Illinois, admits that the concept of phylogeny has lost its meaning in the face of molecular findings in this way. No consistent organismal phylogeny has emerged from the many individual protein phylogenies so far produced. Phylogenetic incongruities can be seen everywhere in the universal tree, from its root to the major branchings within and among the various groups, to the makeup of the primary groupings themselves. 296 The fact that results of molecular comparisons are not in favor of, but rather opposed to, the theory of evolution is also admitted in an article called Is It Time to Uproot the Tree of Life? Published in Science in 1999. This article by Elizabeth Pennisi states that the genetic analyses and comparisons carried out by Darwinist biologists in order to shed light on the tree of life actually yielded directly opposite results, and goes on to say that new data are muddying the evolutionary picture. A year ago, biologists looking over newly sequenced genomes from more than a dozen microorganisms thought these data might support the accepted plot lines of life's early history. But what they saw confounded them. Comparisons of the genomes then available not only didn't clarify the picture of how life's major groupings evolved, they confused it. And now, with an additional 8 microbial sequences in hand, the situation has gotten even more confusing. Many evolutionary biologists had thought they could roughly see the beginnings of life's 3 kingdoms. When full DNA sequences opened the way to comparing other kinds of genes, researchers expected that they would simply add detail to this tree. But nothing could be further from the truth, says Claire Fraser, head of the Institute for Genomic Research, Tigre, in Rockville, Maryland. Instead, the comparisons have yielded many versions of the tree of life that differ from the RNA tree and conflict with each other as well. 297 In short, as molecular biology advances, the homology concept loses more ground. Comparisons that have been made of proteins, RNAs and genes revealed that creatures which are allegedly close relatives according to the theory of evolution are actually totally distinct from each other. A 1996 study using 88 protein sequences grouped rabbits with primates instead of rodents. A 1998 analysis of 13 genes in 19 animal species placed sea urchins among the chordates, and another 1998 study based on 12 proteins put cows closer to whales than to horses. As life is investigated on a molecular basis, the homology hypotheses of the evolutionary theory collapse one by one. Molecular biologist Jonathan Wells sums up the situation in 2000 in this way: inconsistencies among trees based on different molecules, and the bizarre trees that result from some molecular analyses, have now plunged molecular phylogeny into a crisis. 298 But in that case, what kind of scientific explanation can be given for similar structures in living things? The answer to that question was given before Darwin's theory of evolution came to dominate the world of science. Men of science such as Carl Linnaeus and Richard Owen, who first raised the question of similar organs in living creatures, saw these organs as examples of common creation. In other words, similar organs or similar genes resemble each other not because they have evolved by chance from a common ancestor, but because they have been created to perform a particular function. Modern scientific discoveries show that the claim that similarities in living things are due to descent from a common ancestor is not valid, and that the only rational explanation for such similarities is common creation. Chapter 10. Immunity, vestigial organs and embryology. In the preceding sections, we examined the inconsistencies and difficulties the theory of evolution finds itself in the fields of paleontology and molecular biology in the light of scientific proof and discoveries. In this chapter, we shall be considering some biological facts presented as evidence for the theory in evolutionist sources. In contrast to widespread belief, these facts show that there is actually no scientific discovery that supports the theory of evolution. Bacterial resistance to antibiotics. One of the biological concepts that evolutionists try to present as evidence for their theory is the resistance of bacteria to antibiotics. Many evolutionist sources mention antibiotic resistance as an example of the development of living things by advantageous mutations. A similar claim is also made for the insects which build immunity to insecticides such as DDT. However, evolutionists are mistaken on this subject too. Antibiotics are killer molecules that are produced by microorganisms to fight other microorganisms. The first antibiotic was penicillin, discovered by Alexander Fleming in 1928. Fleming realized that mold produced a molecule that killed the Staphylococcus bacterium, and this discovery marked a turning point in the world of medicine. Antibiotics derived from microorganisms were used against bacteria and the results were successful. Soon, something new was discovered. Bacteria build immunity to antibiotics over time. The mechanism works like this: a large proportion of the bacteria that are subjected to antibiotics die, but some others, which are not affected by that antibiotic, replicate rapidly and soon make up the whole population. Thus, the entire population becomes immune to antibiotics. Evolutionists try to present this as the evolution of bacteria by adapting to conditions. The truth, however, is very different from the superficial evolutionary interpretation. One of the scientists who has done the most detailed research into this subject is the Israeli biophysicist Leigh Spetner, who is also known for his book Not by Chance, published in 1997. Spetner maintains that the immunity of bacteria comes about by two different mechanisms, but neither of them constitutes evidence for the theory of evolution. These two mechanisms are, 1, the transfer of resistance genes already extant in bacteria. 2, the building of resistance as a result of losing genetic data because of mutation. Professor Spetner explains the first mechanism in an article published in 2001. Some microorganisms are endowed with genes that grant resistance to these antibiotics. This resistance can take the form of degrading the antibiotic molecule or of ejecting it from the cell. T organisms having these genes can transfer them to other bacteria, making them resistant as well. Although the resistance mechanisms are specific to a particular antibiotic, most pathogenic bacteria have succeeded in accumulating several sets of genes granting them resistance to a variety of antibiotics. 299 Spetner then goes on to say that this is not evidence for evolution. The acquisition of antibiotic resistance in this manner is not the kind that can serve as a prototype for the mutations needed to account for evolution. The genetic changes that could illustrate the theory must not only add information to the bacterium's genome, they must add new information to the biocosm. The horizontal transfer of genes only spreads around genes that are already in some species. 300 So, we cannot talk of any evolution here, because no new genetic information is produced. Genetic information that already exists is simply transferred between bacteria. The second type of immunity, which comes about as a result of mutation, is not an example of evolution either. Spetner writes, A microorganism can sometimes acquire resistance to an antibiotic through a random substitution of a single nucleotide. Streptomycin, which was discovered by Selman Waksman and Albert Schatz and first reported in 1944, is an antibiotic against which bacteria can acquire resistance in this way. But although the mutation they undergo in the process is beneficial to the microorganism in the presence of streptomycin, it cannot serve as a prototype for the kind of mutations needed by NDT, Neo-Darwinian theory. The type of mutation that grants resistance to streptomycin is manifest in the ribosome and degrades its molecular match with the antibiotic molecule. 301 In his book Not by Chance, Spettner likens this situation to the disturbance of the key-lock relationship. Streptomycin, just like a key that perfectly fits in a lock, clutches onto the ribosome of a bacterium and inactivates it. Mutation, on the other hand, decomposes the ribosome, thus preventing streptomycin from holding onto the ribosome. Although this is interpreted as bacteria developing immunity against streptomycin, this is not a benefit for the bacteria but rather a loss for it. Spettner writes, it turns out that this degradation is a loss of specificity and therefore a loss of information. The main point is that evolution cannot be achieved by mutations of this sort, no matter how many of them there are. Evolution cannot be built by accumulating mutations that only degrade specificity. 302 To sum up, a mutation impinging on a bacterium's ribosome makes that bacterium resistant to streptomycin. The reason for this is the decomposition of the ribosome by mutation. That is, no new genetic information is added to the bacterium. On the contrary, the structure of the ribosome is decomposed, that is to say, the bacterium becomes disabled. Also, it has been discovered that the ribosome of the mutated bacterium is less functional than that of a normal bacterium, since this disability prevents the antibiotic from attaching onto the ribosome. Antibiotic resistance develops. Finally, there is no example of mutation that develops the genetic information. Evolutionists, who want to present antibiotic resistance as evidence for evolution, treat the issue in a very superficial way and are thus mistaken. The same situation holds true for the immunity that insects develop to DDT and similar insecticides. In most of these instances, immunity genes that already exist are used. The evolutionary biologist Francisco Ayala admits this fact, saying, the genetic variants required for resistance to the most diverse kinds of pesticides were apparently present in every one of the populations exposed to these man-made compounds. 303 Some other examples explained by mutation, just as with the ribosome mutation mentioned above, are phenomena that cause genetic information deficit in insects. In this case, it cannot be claimed that the immunity mechanisms in bacteria and insects constitute evidence for the theory of evolution. That is because the theory of evolution is based on the assertion that living things develop through mutations. However, Spettner explains that neither antibiotic immunity nor any other biological phenomena indicate such an example of mutation. The mutations needed for macroevolution have never been observed. No random mutations that could represent the mutations required by neo-Darwinian theory that have been examined on the molecular level have added any information. The question I address is, are the mutations that have been observed the kind the theory needs for support? The answer turns out to be no! 304 The myth of vestigial organs. For a long time, the concept of the vestigial organs appeared frequently in evolutionist literature as evidence of evolution. Eventually, it was silently put to rest when this was proved to be invalid. But some evolutionists still believe in it, and from time to time someone will try to advance vestigial organs as important evidence of evolution. The notion of vestigial organs was first put forward a century ago. As evolutionists would have it, there existed in the bodies of some creatures a number of non-functional organs. These had been inherited from progenitors and had gradually become vestigial from lack of use. The whole assumption is quite unscientific, and is based entirely on insufficient knowledge. These non-functional organs were in fact organs whose functions had not yet been discovered. The best indication of this was the gradual yet substantial decrease in evolutionists' long list of the vestigial organs. S.R. Scatting, an evolutionist himself, concurred with this fact in his article Can Vestigial Organs Constitute Evidence for Evolution? Published in the journal Evolutionary Theory. Since it is not possible to unambiguously identify useless structures, and since the structure of the argument used is not scientifically valid, I conclude that vestigial organs provide no special evidence for the theory of evolution. 305 The list of the vestigial organs that was made by the German anatomist Harun Yahya in 1895 included approximately 100 organs, including the appendix and coccyx. As science progressed, it was discovered that all of the organs in Yahya's list in fact had very important functions. For instance, it was discovered that the appendix, which was supposed to be a vestigial organ, was in fact a lymphoid organ that fought infections in the body. This fact was made clear in 1997. Other bodily organs and tissues, the thymus, liver, spleen, appendix, bone marrow, and small collections of lymphatic tissue such as the tonsils in the throat and Peyer's patch in the small intestine are also part of the lymphatic system. They too help the body fight infection. 306 It was also discovered that the tonsils, which were included in the same list of vestigial organs, had a significant role in protecting the throat against infections, particularly until adolescence. It was found that the coccyx at the lower end of the vertebral column supports the bones around the pelvis and is the convergence point of some small muscles, and for this reason, it would not be possible to sit comfortably without a coccyx. In the years that followed, it was realized that the thymus triggered the immune system in the human body by activating the T cells, that the pineal gland was in charge of the secretion of some important hormones such as melatonin, which inhibits secretion of luteinizing hormone, that the thyroid gland was effective in providing steady growth in babies and children and in metabolism and body activity, and that the pituitary gland controlled skeletal growth and the proper functioning of the thyroid, adrenals, and reproductive glands. All of these were once considered to be vestigial organs. Finally, the semilunar fold in the eye, which was referred to as a vestigial organ by Darwin, has been found in fact to be in charge of cleansing and lubricating the eyeball. There was a very important logical error in the evolutionist claim regarding vestigial organs. As we have just seen, this claim was that the vestigial organs in living things were inherited from their ancestors. However, some of the alleged vestigial organs are not found in the species alleged to be the ancestors of human beings. For example, the appendix does not exist in some ape species that are said to be ancestors of man. The famous biologist H. Enoch, who challenged the theory of vestigial organs, expressed this logical error as follows: apes possess an appendix, whereas their less immediate relatives, the lower apes, do not, but it appears again among the still lower mammals such as the opossum. How can the evolutionists account for this? 307 Beside all of this, the claim that an organ which is not used atrophies and disappears over time carries a logical inconsistency within it. Darwin was aware of this inconsistency, and made the following confession in The Origin of Species: there remains, however, this difficulty. After an organ has ceased being used, and has become in consequence much reduced, how can it be still further reduced in size until the merest vestige is left, and how can it be finally quite obliterated? It is scarcely possible that disuse can go on producing any further effect after the organ has once been rendered functionless. Some additional explanation is here requisite which I cannot give. 308 Simply put, the scenario of the vestigial organs put forward by evolutionists contains a number of serious logical flaws, and has in any case been proven to be scientifically untrue. There exists not one inherited vestigial organ in the human body. Yet another blow to vestigial organs, the leg of the horse. The latest blow to the myth of vestigial organs comes from a recent study on the leg of the horse. In an article in the December 27, 2001 issue of the journal Nature, titled Biomechanics, Damper for Bad Vibrations, it is noted that some muscle fibers in the legs of horses seem to be evolutionary leftovers with no function. But in fact they may act to damp damaging vibrations generated in the leg as the horse runs. The article reads as follows: horses and camels have muscles in their legs with tendons more than 600 mm long connected to muscle fibers less than 6 mm long. Such short muscles can change length only by a few millimeters as the animal moves, and seem unlikely to be of much use to large mammals. The tendons function as passive springs, and it has been assumed that the short muscle fibers are redundant, the remnants of longer fibers that have lost their function over the course of evolution. But Wilson and colleagues argue that these fibers might protect bones and tendons from potentially damaging vibrations. Their experiments show that short muscle fibers can damp the damaging vibrations following the impact of a foot on the ground. When the foot of a running animal hits the ground, the impact sets the leg vibrating. The frequency of the vibrations is relatively high, for example, 30 to 40 Hz in horses. So many cycles of vibration would occur while the foot was on the ground if there were no damping. The vibrations might cause damage, because bone and tendon are susceptible to fatigue failure. Fatigue in bones and tendons is the accumulation of damage resulting from repeated application of stresses. Bone fatigue is responsible for the stress fractures suffered by both human athletes and race horses, and tendon fatigue may explain at least some cases of tendinitis. Wilson et al. Suggest that the very short muscle fibers protect both bones and tendons from fatigue damage by damping out vibrations. 309 In short, a closer look at the anatomy of the horse revealed that the structures that have been considered as non-functional by evolutionists have very important functions. In other words, scientific progress demonstrated that what was considered to be evidence for evolution is in fact evidence for creation. Evolutionists should be objective and evaluate scientific findings reasonably. The Nature article comments as follows: Wilson et al. Have found an important role for a muscle that seemed to be the relic of a structure that had lost its function in the course of evolution. Their work makes us wonder whether other vestiges, such as the human appendix, are as useless as they seem. 310 This is not surprising. The more we learn about nature, the more we see the evidence for creation. As Michael notes, the conclusion of design comes not from what we do not know, but from what we have learned over the past 50 years. 311 And Darwinism turns out to be an argument from ignorance. The recapitulation misconception, what used to be called the recapitulation theory, has long been eliminated from scientific literature, but it is still being presented as a scientific reality by some evolutionist publications. The term recapitulation is a condensation of the dictum ontogeny recapitulates phylogeny, put forward by the evolutionary biologist Ernst Haeckel at the end of the 19th century. This theory of Haeckel's postulates that living embryos re-experience the evolutionary process that their pseudo-ancestors underwent. He theorized that during its development in its mother's womb, the human embryo first displayed the characteristics of a fish, and then those of a reptile, and finally those of a human. It has since been proven that this theory is completely bogus. It is now known that the gills that supposedly appear in the early stages of the human embryo are in fact the initial phases of the middle ear canal, parathyroid, and thymus. That part of the embryo that was likened to the egg yolk pouch turns out to be a pouch that produces blood for the infant. The part that was identified as a tail by Haeckel and his followers is in fact the backbone, which resembles a tail only because it takes shape before the legs do. These are universally acknowledged facts in the scientific world, and are accepted even by evolutionists themselves. Two leading Darwinists, George Gaylord Simpson and W. Beck, have admitted Haeckel misstated the evolutionary principle involved. It is now firmly established that ontogeny does not repeat phylogeny. 312 The following was written in an article in New Scientist dated October 16, 1999. Haeckel called this the biogenetic law, and the idea became popularly known as recapitulation. In fact, Haeckel's strict law was soon shown to be incorrect. For instance, the early human embryo never has functioning gills like a fish, and never passes through stages that look like an adult reptile or monkey. 313 In an article published in American Scientist, we read, "Surely the biogenetic law is as dead as a doornail." It was finally exorcised from biology textbooks in the 50s. As a topic of serious theoretical inquiry, it was extinct in the 20s. 314 Another interesting aspect of recapitulation was Ernst Haeckel himself, a faker who falsified his drawings in order to support the theory he advanced. Haeckel's forgeries purported to show that fish and human embryos resembled one another. When he was caught out, the only defense he offered was that other evolutionists had committed similar offenses. After this compromising confession of forgery, I should be obliged to consider myself condemned and annihilated if I had not the consolation of seeing side by side with me in the prisoner's dock hundreds of fellow culprits, among them many of the most trusted observers and most esteemed biologists. The great majority of all the diagrams in the best biological textbooks, treatises and journals would incur in the same degree the charge of forgery, for all of them are inexact, and are more or less doctored, schematized and constructed. 315 In the September 5, 1997, edition of the well-known scientific journal Science, an article was published revealing that Haeckel's embryo drawings were the product of a deception. The article, called Haeckel's Embryos, Fraud Rediscovered, had this to say: the impression they, Haeckel's drawings, give, that the embryos are exactly alike, is wrong, says Michael Richardson, an embryologist at St George's Hospital Medical School in London. So he and his colleagues did their own comparative study, re-examining and photographing embryos roughly matched by species and age with those Haeckel drew. Lo and behold, the embryos often looked surprisingly different, Richardson reports in the August issue of Anatomy and Embryology. 316 Science explained that, in order to be able to show the embryos as similar, Haeckel deliberately removed some organs from his drawings or else added imaginary ones. Later in this same article, the following information was revealed: not only did Haeckel add ornament features, Richardson and his colleagues report, but he also fudged the scale to exaggerate similarities among species, even when there were tenfold differences in size. Haeckel further blurred differences by neglecting to name the species in most cases, as if one representative was accurate for an entire group of animals. In reality, Richardson and his colleagues note, even closely related embryos such as those of fish vary quite a bit in their appearance and developmental pathway. Haeckel's drawings, looks like it's turning out to be one of the most famous fakes in biology, Richardson concludes. 317 The Science article goes on to discuss how Haeckel's confessions on this subject were covered up from the beginning of the last century, and how the fake drawings began to be presented in textbooks as scientific fact. Haeckel's confession got lost after his drawings were subsequently used in a 1901 book called Darwin and After Darwin and reproduced widely in English-language biology texts. 318 In short, the fact that Haeckel's drawings were falsified had already emerged in 1901, but the whole world of science continued to be deceived by them for a century. Chapter 11 The Origin of Plants. Life on Earth is divided into 5, or sometimes 6, kingdoms by scientists. We have so far concentrated mainly on the greatest kingdom, that of animals. In the preceding chapters, we considered the origin of life itself, studying proteins, genetic information, cell structure and bacteria, issues that are related with 2 other kingdoms, prokaryote and protista. But at this point there is another important matter we need to concentrate on: the origin of the plant kingdom, Plantae. We find the same picture in the origin of plants as we met when examining the origin of animals. Plants possess exceedingly complex structures, and it is not possible for these to come about by chance effects and for them to evolve into one another. The fossil record shows that the different classes of plants emerged all of a sudden in the world, each with its own particular characteristics, and with no period of evolution behind it. The origin of the plant cell. Like animal cells, plant cells belong to the type known as eukaryotic. The most distinctive feature of these is that they have a cell nucleus, and the DNA molecule in which their genetic information is encoded lies within this nucleus. On the other hand, some single-celled creatures such as bacteria have no cell nucleus, and the DNA molecule is free inside the cell. This second type of cell is called prokaryotic. This type of cell structure, with free DNA unconfined within a nucleus, is an ideal design for bacteria, as it makes possible the very important process from the bacterial point of view of plasmid transfer, that is, the transfer of DNA from cell to cell. Because the theory of evolution is obliged to arrange living things in a sequence from primitive to complex, it assumes that prokaryotic cells are primitive, and that eukaryotic cells evolved from them. Before moving to the invalidity of this claim, it will be useful to demonstrate that prokaryotic cells are not at all primitive. A bacterium possesses some 2,000 genes, each gene contains about 100 letters, links. This means that the information in a bacterium's DNA is some 200,000 letters long. According to this calculation, the information in the DNA of one bacterium is equivalent to 20 novels, each of 10,000 words. 319 Any change in the information in the DNA code of a bacterium would be so deleterious as to ruin the bacterium's entire working system. As we have seen, a fault in a bacterium's genetic code means that the working system will go wrong, that is, the cell will die. Alongside this sensitive structure, which defies chance changes, the fact that no intermediate form between bacteria and eukaryotic cells has been found makes the evolutionist claim unfounded. For example, the famous Turkish evolutionist Professor Ali Demirsoy confesses the groundlessness of the scenario that bacterial cells evolved into eukaryotic cells, and then into complex organisms made up of these cells. One of the most difficult stages to be explained in evolution is to scientifically explain how organelles and complex cells developed from these primitive creatures. No transitional form has been found between these two forms. One and multi-celled creatures carry all this complex structure, and no creature or group has yet been found with organelles of a simpler construction in any way, or which are more primitive. In other words, the organelles carried forward have developed just as they are. They have no simple and primitive forms. 321 wonders, what is it that encourages Professor Ali Demirsoy, a loyal adherent of the theory of evolution, to make such an open admission? The answer to this question can be given quite clearly when the great structural differences between bacteria and plant cells are examined. These are: 1. While the walls of bacterial cells are formed of polysaccharide and protein, the walls of plant cells are formed of cellulose, a totally different structure. 2. While plant cells possess many organelles, covered in membranes and possessing very complex structures, bacterial cells lack typical organelles. In bacterial cells there are just freely moving tiny ribosomes. But the ribosomes in plant cells are larger and are attached to the cell membrane. Furthermore, protein synthesis takes place by different means in the two types of ribosomes. 3. The DNA structures in plant and bacterial cells are different. 4. The DNA molecule in plant cells is protected by a double-layered membrane, whereas the DNA in bacterial cells stands free within the cell. 5. The DNA molecule in bacterial cells resembles a closed loop, in other words, it is circular. In plants, the DNA molecule is linear. 6. The DNA molecule in bacterial cells carries information belonging to just one cell, but in plant cells the DNA molecule carries information about the whole plant. For example, all the information about a fruit-bearing tree's roots, stem, leaves, flowers, and fruit are all found separately in the DNA in the nucleus of just one cell. 7. Some species of bacteria are photosynthetic, in other words, they carry out photosynthesis. But unlike plants, in photosynthetic bacteria, cyanobacteria, for instance, there is no chloroplast containing chlorophyll and photosynthetic pigments. Rather, these molecules are buried in various membranes all over the cell. 8. The biochemistry of messenger RNA formation in prokaryotic, bacterial, cells and in eukaryotic, including plant and animal, cells are quite different from one another. 321 Messenger RNA plays a vital role for the cell to live. But although messenger RNA assumes the same vital role in both prokaryotic cells and in eukaryotic cells, their biochemical structures are different. J. Darnell wrote the following in an article published in Science: the differences in the biochemistry of messenger RNA formation in eukaryotes compared to prokaryotes are so profound as to suggest that sequential prokaryotic to eukaryotic cell evolution seems unlikely. 322 The structural differences between bacterial and plant cells, of which we have seen a few examples above, lead evolutionist scientists to another dead end. Although plant and bacterial cells have some aspects in common, most of their structures are quite different from one another. In fact, since there are no membrane-surrounded organelles or a cytoskeleton, the internal network of protein filaments in microtubules, in bacterial cells, the presence of several very complex organelles and cell organization in plant cells totally invalidates the claim that the plant cell evolved from the bacterial cell. Biologist Ollie Demersoy openly admits this, saying, "Complex cells never developed from primitive cells by a process of evolution." 323 The antisymbiosis hypothesis and its invalidity. The impossibility of plant cells having evolved from a bacterial cell has not prevented evolutionary biologists from producing speculative hypotheses. But experiments disprove these. 324 The most popular of these is the antisymbiosis hypothesis. This hypothesis was put forward by Lynn Margulis in 1970 in her book The Origin of Eukaryotic Cells. In this book, Margulis claimed that as a result of their communal and parasitic lives, bacterial cells turned into plant and animal cells. According to this theory, plant cells emerged when a photosynthetic bacterium was swallowed by another bacterial cell. The photosynthetic bacterium evolved inside the parent cell into a chloroplast. Lastly, organelles with highly complex structures such as the nucleus, the Golgi apparatus, the endoplasmic reticulum, and ribosomes evolved, in some way or other. Thus, the plant cell was born. As we have seen, this thesis of the evolutionists is nothing but a work of fantasy. Unsurprisingly, it was criticized by scientists who carried out very important research into the subject on a number of grounds. We can cite D. Lloyd 325, M. Gray and W. Doolittle 326, and R. Ruff and H. Mahler as examples of these. The antisymbiosis hypothesis is based on the fact that the mitochondria of animal cells and the chloroplasts of plant cells contain their own DNA, separate from the DNA in the nucleus of the parent cell. So, on this basis, it is suggested that mitochondria and chloroplasts were once independent, free-living cells. However, when chloroplasts are studied in detail, it can be seen that this claim is inconsistent. A number of points invalidate the antisymbiosis hypothesis. 1. If chloroplasts, in particular, were once independent cells, then there could only have been one outcome if one were swallowed by a larger cell, namely, it would have been digested by the parent cell and used as food. This must be so, because even if we assume that the parent cell in question took such a cell into itself from the outside by mistake, instead of intentionally ingesting it as food, nevertheless, the digestive enzymes in the parent cell would have destroyed it. Of course, some evolutionists have gotten around this obstacle by saying, the digestive enzymes had disappeared. But this is a clear contradiction, because if the cell's digestive enzymes had disappeared, then the cell would have died from lack of nutrition. 2. Again, let us assume that all the impossible happened and that the cell which is claimed to have been the ancestor of the chloroplast was swallowed by the parent cell. In this case we are faced with another problem. The blueprints of all the organelles inside the cell are encoded in the DNA. If the parent cell were going to use other cells it swallowed as organelles, then it would be necessary for all of the information about them to be already present and encoded in its DNA. The DNA of the swallowed cells would have to possess information belonging to the parent cell. Not only is such a situation impossible, the two complements of DNA belonging to the parent cell and the swallowed cell would also have to become compatible with each other afterwards, which is also clearly impossible. 3. There is great harmony within the cell which random mutations cannot account for. There are more than just one chloroplast and one mitochondrion in a cell. Their number rises or falls according to the activity level of the cell, just like with other organelles. The existence of DNA in the bodies of these organelles is also of use in reproduction. As the cell divides, all of the numerous chloroplasts divide too, and the cell division happens in a shorter time and more regularly. For chloroplasts are energy generators of absolutely vital importance to the plant cell. If these organelles did not produce energy, many of the cell's functions would not work, which would mean that the cell could not live. These functions, which are so important to the cell, take place with proteins synthesized in the chloroplasts. But the chloroplast's own DNA is not enough to synthesize these proteins. The greater part of the proteins are synthesized using the parent DNA in the cell nucleus. 327 While the situation envisioned by the endosymbiosis hypothesis is occurring through a process of trial and error, what effects would this have on the DNA of the parent cell? As we have seen, any change in a DNA molecule definitely does not result in a gain for that organism. On the contrary, any such mutation would certainly be harmful. In his book The Routes of Life, Elon B. Hoagland explains the situation. You'll recall we learned that almost always a change in an organism's DNA is detrimental to it, that is, it leads to a reduced capacity to survive. By way of analogy, random additions of sentences to the plays of Shakespeare are not likely to improve them. The principle that DNA changes are harmful by virtue of reducing survival chances applies whether a change in DNA is caused by a mutation or by some foreign genes we deliberately add to it. 328 The claims put forward by evolutionists are not based on scientific experiments, because no such thing as one bacterium swallowing another one has ever been observed. In his review of a later book by Margulis, Symbiosis in Cell Evolution, molecular biologist P. Whitfield describes the situation. Prokaryotic endocytosis is the cellular mechanism on which the whole of SCT serial endosymbiotic theory, presumably rests. If one prokaryote could not engulf another, it is difficult to imagine how endosymbioses could be set up. Unfortunately for Margulis and SCT, no modern examples of prokaryotic endocytosis or endosymbiosis exist. 329 The origin of photosynthesis. Another matter regarding the origin of plants which puts the theory of evolution into a terrible quandary is the question of how plant cells began to carry out photosynthesis. Photosynthesis is one of the fundamental processes of life on Earth. By means of the chloroplasts inside them, plant cells produce starch by using water, carbon dioxide and sunlight. Animals are unable to produce their own nutrients and must use the starch from plants for food instead. For this reason, photosynthesis is a basic condition for complex life. An even more interesting side of the matter is the fact that this complex process of photosynthesis has not yet been fully understood. Modern technology has not yet been able to reveal all of its details, let alone reproduce it. How is it that evolutionists believe such a complex process as photosynthesis is the product of natural and random processes? According to the evolution scenario, in order to carry out photosynthesis, plant cells swallowed bacterial cells which could photosynthesize and turn them into chloroplasts. So, how did bacteria learn to carry out such a complex process as photosynthesis? And why had they not begun to carry out such a process before then? As with other questions, the scenario has no scientific answer to give. Have a look at how an evolutionist publication answers the question. The heterotroph hypothesis suggests that the earliest organisms were heterotrophs that fed on a soup of organic molecules in the primitive ocean. As these first heterotrophs consumed the available amino acids, proteins, fats, and sugars, the nutrient soup became depleted and could no longer support a growing population of heterotrophs. Organisms that could use an alternate source of energy would have had a great advantage. Consider that Earth was, and continues to be, flooded with solar energy that actually consists of different forms of radiation. Ultraviolet radiation is destructive, but visible light is energy-rich and undestructive. Thus, as organic compounds became increasingly rare, an already present ability to use visible light as an alternate source of energy might have enabled such organisms and their descendants to survive. 330 The book Life on Earth, another evolutionist source, tries to explain the emergence of photosynthesis, The bacteria fed initially on the various carbon compounds that had taken so many millions of years to accumulate in the primordial seas. But as they flourished, so this food must have become scarcer. Any bacterium that could tap a different source of food would obviously be very successful, and eventually some did. Instead of taking ready-made food from their surroundings, they began to manufacture their own within their cell walls, drawing the necessary energy from the sun. 331 In short, evolutionist sources say that photosynthesis was in some way coincidentally discovered by bacteria, even though man, with all his technology and knowledge, has been unable to do so. These accounts, which are no better than fairy tales, have no scientific worth. Those who study the subject in a bit more depth will accept that photosynthesis is a major dilemma for evolution. Professor Ali Demirsoy makes the following admission: for instance, photosynthesis is a rather complicated event, and it seems impossible for it to emerge in an organelle inside a cell, because it is impossible for all the stages to have come about at once, and it is meaningless for them to have emerged separately. 332 The German biologist Hoymar von Wittfurth says that photosynthesis is a process that cannot possibly be learned. No cell possesses the capacity to learn a process in the true sense of the word. It is impossible for any cell to come by the ability to carry out such functions as respiration or photosynthesis, neither when it first comes into being, nor later in life. 333 Since photosynthesis cannot develop as the result of chance, and cannot subsequently be learned by a cell, it appears that the first plant cells that lived on the earth were specially created to carry out photosynthesis. In other words, plants were created by Allah with the ability to photosynthesize. The origin of algae. The theory of evolution hypothesizes that single-celled plant-like creatures, whose origins it is unable to explain, came in time to form algae. The origin of algae goes back to very remote times. So much so, that fossil algae remains from 3.1 to 3.4 million years old have been found. The interesting thing is that there is no structural difference between these extraordinarily ancient living things and specimens living in our own time. An article published in Science News says, both blue-green algae and bacteria fossils dating back 3.4 billion years have been found in rocks from Africa. Even more intriguing, the pleuropapsilin algae turned out to be almost identical to modern pleuropapsilin algae at the family and possibly even at the generic level. 334 The German biologist Hoymar von Wittfurth makes this comment on the complex structure of so-called primitive algae. The oldest fossils so far discovered are objects fossilized in minerals which belong to blue-green algae, more than 3 billion years old. No matter how primitive they are, they still represent rather complicated and expertly organized forms of life. 335 Evolutionary biologists consider that the algae in question gave rise over time to other marine plants and moved to the land some 450 million years ago. However, just like the scenario of animals moving from water onto the land, the idea that plants moved from water to the land is another fantasy. Both scenarios are invalid and inconsistent. Evolutionist sources usually try to gloss over the subject with such fantastical and unscientific comments as algae in some way moved onto the land and adapted to it. But there are a large number of obstacles that make this transition quite impossible. Let us have a short look at the most important of them. 1. The danger of drying out. For a plant which lives in water to be able to live on land, its surface has first of all to be protected from water loss. Otherwise the plant will dry out. Land plants are provided with special systems to prevent this from happening. There are very important details in these systems. For example, this protection must happen in such a way that important gases such as oxygen and carbon dioxide are able to leave and enter the plant freely. At the same time, it is important that evaporation be prevented. If a plant does not possess such a system, it cannot wait millions of years to develop one. In such a situation, the plant will soon dry up and die. 2. Feeding. Marine plants take the water and minerals they need directly from the water they are in. For this reason, Any algae which tried to live on land would have a food problem. They could not live without resolving it. 3. Reproduction. Algae, with their short lifespan, cannot possibly reproduce on land, because, as in all their functions, algae also use water to disperse their reproductive cells. In order to be able to reproduce on land, they would need to possess multicellular reproductive cells like those of land plants, which are covered by a protective layer of cells. Lacking these, any algae which found themselves on land would be unable to protect their reproductive cells from danger. For protection from oxygen, any algae which arrived on land would have taken in oxygen in a decomposed form up until that point. According to the evolutionist scenario, now they would have to take in oxygen in a form they had never encountered before, in other words, directly from the atmosphere. As we know, under normal conditions the oxygen in the atmosphere has a poisoning effect on organic substances. Living things which live on land possess systems which stop them being harmed by it. But algae are marine plants, which means they do not possess the enzymes to protect them from the harmful effects of oxygen. So, as soon as they arrived on land, it would be impossible for them to avoid these effects. Neither is there any question of their waiting for such a system to develop, because they could not survive on land long enough for that to happen. There is yet another reason why the claim that algae moved from the ocean to the land inconsistent, namely, the absence of a natural agent to make such a transition necessary. Let us imagine the natural environment of algae 450 million years ago. The waters of the sea offer them an ideal environment. For instance, the water isolates and protects them from extreme heat, and offers them all kinds of minerals they need. And, at the same time, they can absorb the sunlight by means of photosynthesis and make their own carbohydrates, sugar and starch, by carbon dioxide, which dissolves in the water. For this reason, there is nothing the algae lack in the ocean, and therefore no reason for them to move to the land, where there is no selective advantage for them, as the evolutionists put it. All of this shows that the evolutionist hypothesis that algae emerged onto the land and formed land plants is completely unscientific. The origin of angiosperms. When we examine the fossil history and structural features of plants that live on land, another picture emerges which fails to agree with evolutionist predictions. There is no fossil series to confirm even one branch of the evolutionary tree of plants that you will see in almost any biological textbook. Most plants possess abundant remains in the fossil record, but none of these fossils is an intermediate form between one species and another. They are all specially and originally created as completely distinct species, and there are no evolutionary links between them. As the evolutionary paleontologist T.C. Olson accepted, many new groups of plants and animals suddenly appear, apparently without any close ancestors. 336. The botanist Chester A. Arnold, who studies fossil plants at the University of Michigan, makes the following comment: "It has long been hoped that extinct plants will ultimately reveal some of the stages through which existing groups have passed during the course of their development, but it must be freely admitted that this aspiration has been fulfilled to a very slight extent, even though paleobotanical research has been in progress for more than 100 years. 337. Arnold accepts that paleobotany, the science of plant fossils, has produced no results in support of evolution. We have not been able to track the phylogenetic history of a single group of modern plants from its beginning to the present. 338. The fossil discoveries which most clearly deny the claims of plant evolution are those of flowering plants, or angiosperms, to give them their scientific name. These plants are divided into 43 separate families, each one of which emerges suddenly, leaving no trace of any primitive transitional form behind it in the fossil record. This was realized in the 19th century, and for this reason Darwin described the origin of angiosperms as an abominable mystery. All the research carried out since Darwin's time has simply added to the amount of discomfort this mystery causes. In his book The Paleobiology of Angiosperm Origins, the evolutionary paleobotanist N. F. Hughes makes this admission. With few exceptions of detail, however, The failure to find a satisfactory explanation has persisted, and many botanists have concluded that the problem is not capable of solution, by use of fossil evidence. 339 In his book The Evolution of Flowering Plants, Daniel Axelrod says this about the origin of flowering plants: the ancestral group that gave rise to angiosperms has not yet been identified in this fossil record, and no living angiosperm points to such an ancestral alliance. 340 All this leads us to just one conclusion: like all living things, plants were also created. From the moment they first emerged, all their mechanisms have existed in a finished and complete form. Terms such as development over time, changes dependent on coincidences, and adaptations which emerged as a result of need, which one finds in the evolutionist literature, have no truth in them at all and are scientifically meaningless. Chapter 12. Irreducible Complexity. One of the most important concepts that one must employ when questioning Darwinist theory in the light of scientific discoveries is without a doubt the criterion that Darwin himself employed. In The Origin of Species, Darwin put forward a number of concrete criteria suggesting how his theory might be tested and, if found wanting, disproved. Many passages in his book begin, "If my theory be true," and in these Darwin describes the discoveries his theory requires. One of the most important of these criteria concerns fossils and transitional forms. In earlier chapters, we examine how these prophecies of Darwin's did not come true, and how, on the contrary, the fossil record completely contradicts Darwinism. In addition to these, Darwin gave us another very important criterion by which to test his theory. This criterion is so important, Darwin wrote, that it could cause his theory to be absolutely broken down, if it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down. But I can find out no such case. 341 We must examine Darwin's intention here very carefully. As we know, Darwinism explains the origin of life with two unconscious natural mechanisms: natural selection and random changes, in other words, mutations. According to Darwinist theory, these two mechanisms led to the emergence of the complex structure of living cells, as well as the anatomical systems of complex living things, such as eyes, ears, wings, lungs, bats and millions of other complex systems. However, how is it that these systems, which possess complex structures, can be considered the products of two unconscious natural effects? At this point, the concept Darwinism implies is that of reducibility. It is claimed that these systems can be reduced to very basic states, and that they may have then developed by stages. Each stage gives a living thing a little more advantage, and is therefore chosen through natural selection. Then, later, there will be another small, chance development, and that too will be preferred because it affords an advantage, and the process will go on in this way. As a result of this, according to the Darwinist claim, a species which originally possessed no eyes will come to possess perfect ones, and another species which was formerly unable to fly, will grow wings and be able to do so. This story is explained in a very convincing and reasonable manner in evolutionist sources. But when one reflects on it, a great error appears. The first aspect of this error is a subject we have already studied in earlier pages of this book: mutations are destructive, not constructive. In other words, chance mutations that occur in living creatures do not provide them any advantages, and, furthermore, the idea that they could do this thousands of times, one after the other, is a dream that contradicts all scientific observations. But there is yet another very important aspect to the error. Darwinist theory requires all the stages from one point to another to be individually advantageous. In an evolutionary process from A to Z, for instance, from a wingless creature to a winged one, all the intermediate stages B, C, D, V, W, X, and Y along the way have to provide advantages for the living thing in question. Since it is not possible for natural selection and mutation to consciously pick out their targets in advance, the whole theory is based on the hypothesis that living systems can be reduced to discrete traits that can be added onto the organism in small steps, each of which carries some selective advantage. That is why Darwin said, if it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down. Given the primitive level of science in the 19th century, Darwin may have thought that living things possess a reducible structure. But 20th century discoveries have shown that many systems and organs in living things cannot be reduced to simplicity. This fact, known as irreducible complexity, definitively destroys Darwinism, just as Darwin himself feared. The bacterial flagellum. The most important person to bring the concept of irreducible complexity to the forefront of the scientific agenda is the biochemist Michael J. B. of Lehigh University in the United States. In his book Darwin's Black Box, The Biochemical Challenge to Evolution, published in 1996, he examines the irreducibly complex structure of the cell and a number of other biochemical structures, and reveals that it is impossible to account for these by evolution. According to B, the real explanation of life is creation. B's book was a serious blow to Darwinism. In fact, Peter van Inmelgren, professor of philosophy at the University of Notre Dame, stresses the importance of the book in this manner. If Darwinians respond to this important book by ignoring it, misrepresenting it, or ridiculing it, that will be evidence in favor of the widespread suspicion that Darwinism today functions more as an ideology than as a scientific theory. If they can successfully answer B's arguments, that will be important evidence in favor of Darwinism. 342. One of the interesting examples of irreducible complexity that B gives in his book is the bacterial flagellum. This is a whip-like organ that is used by some bacteria to move about in a liquid environment. This organ is embedded in the cell membrane, and enables the bacterium to move in a chosen direction at a particular speed. Scientists have known about the flagellum for some time. However, its structural details, which have only emerged over the last decade or so, have come as a great surprise to them. It has been discovered that the flagellum moves by means of a very complicated organic motor, and not by a simple vibratory mechanism as was earlier believed. This propeller-like engine is constructed on the same mechanical principles as an electric motor. There are two main parts to it, a moving part, the rotor, and a stationary one, the stator. The bacterial flagellum is different from all other organic systems that produce mechanical motion. The cell does not utilize available energy stored as ATP molecules. Instead, it has a special energy source. Bacteria use energy from the flow of ions across their outer cell membranes. The inner structure of the motor is extremely complex. Approximately 240 distinct proteins go into constructing the flagellum. Each one of these is carefully positioned. Scientists have determined that these proteins carry the signals to turn the motor on or off, form joints to facilitate movements at the atomic scale, and activate other proteins that connect the flagellum to the cell membrane. The models constructed to summarize the working of the system are enough to depict the complicated nature of the system. The complicated structure of the bacterial flagellum is sufficient all by itself to demolish the theory of evolution, since the flagellum has an irreducibly complex structure. If one single molecule in this fabulously complex structure were to disappear, or become defective, the flagellum would neither work nor be of any use to the bacterium. The flagellum must have been working perfectly from the first moment of its existence. This fact again reveals the nonsense in the theory of evolution's assertion of step-by-step development. In fact, not one evolutionary biologist has so far succeeded in explaining the origin of the bacterial flagellum, although a few tried to do so. The bacterial flagellum is clear evidence that even in supposedly primitive creatures there are extraordinary features. As humanity learns more about the details, it becomes increasingly obvious that the organisms considered to be the simplest by the scientists of 19th century, including Darwin, are in fact just as complex as any others. The creation of the human eye. The human eye is a very complex system consisting of the delicate conjunction of some 40 separate components. Let us consider just one of these components, for example, the lens. We do not usually realize it, but the thing that enables us to see things clearly is the constant automatic focusing of the lens. If you wish, you can carry out a small experiment on this subject. Hold your index finger up in the air. Then look at the tip of your finger, then at the wall behind it. Every time you look from your finger to the wall you will feel an adjustment. This adjustment is made by small muscles around the lens. Every time we look at something, these muscles go into action and enable us to see what we are looking at clearly by changing the thickness of the lens and turning it at the right angle to the light. The lens carries out this adjustment every second of our lives, and makes no mistakes. Photographers make the same adjustments in their cameras by hand, and sometimes have to struggle for quite some time to get the right focus. Within the last 10 to 15 years, modern technology has produced cameras which focus automatically, but no camera can focus as quickly and as well as the eye. For an eye to be able to see, the 40 or so basic components which make it up need to be present at the same time and work together perfectly. The lens is only one of these. If all the other components, such as the cornea, iris, pupil, retina, and eye muscles, are all present and functioning properly, but just the eyelid is missing, then the eye will shortly incur serious damage and cease to carry out its function. In the same way, if all the subsystems exist but tear production ceases, then the eye will dry up and go blind within a few hours. The theory of evolution's claim of reducibility loses all meaning in the face of the complex structure of the eye. The reason is that, in order for the eye to function, all its parts need to be present at the same time. It is impossible, of course, for the mechanisms of natural selection and mutation to give rise to the eye's dozens of different subsystems when they can confer no advantage right up until the last stage. Professor Ali Demirsoy accepts the truth of this in these words: "It is rather hard to reply to a third objection. How was it possible for a complicated organ to come about suddenly even though it brought benefits with it? For instance, how did the lens, retina, optic nerve, and all the other parts and vertebrates that play a role in seeing suddenly come about? Because natural selection cannot choose separately between the visual nerve and the retina. The emergence of the lens has no meaning in the absence of a retina. The simultaneous development of all the structures for sight is unavoidable. Since parts that develop separately cannot be used, they will both be meaningless, and also perhaps disappear with time. At the same time, their development altogether requires the coming together of unimaginably small probabilities." 343 What Professor Demirsoy really means by unimaginably small probabilities is basically an impossibility. It is clearly an impossibility for the eye to be the product of chance. Darwin also had a great difficulty in the face of this, and in a letter he even admitted, "I remember well the time when the thought of the eye made me cold all over." 344 In The Origin of Species, Darwin experienced this serious difficulty in the face of the eye's complexity. The only solution he found was in pointing to the simpler eye structure found in some creatures as the origin of the more complex eyes found in others. He claimed that more complex eyes evolved from simpler ones. However, this claim does not reflect the truth. Paleontology shows that living things emerged in the world with their exceedingly complex structures already intact. The oldest known system of sight is the trilobite eye. This 530-million-year-old compound eye structure, which we touched on in an earlier chapter, is an optical marvel which worked with a double lens system. This fact totally invalidates Darwin's assumption that complex eyes evolved from primitive eyes. The irreducible structure of the primitive eye. It remains to be said that the organs described by Darwin as primitive eyes actually possess a complex and irreducible structure that can never be explained by chance. Even in its simplest form, for seeing to happen, some of a creature's cells need to become light-sensitive, that is, they need to possess the ability to transduce this sensitivity to light into electrical signals. A nerve network from these cells to the brain needs to emerge. and a visual center in the brain to evaluate the information has to be formed. It is senseless to propose that all of these things came about by chance, at the same time, and in the same living thing. In his book Harun Yahya v. Bagnaslik, The Theory of Evolution and Bigotry, which he wrote to defend the theory of evolution, the evolutionist writer Samuel Yildirim admits this fact in this way: a large number of mechanisms need to work together for sight, as well as the eye and the mechanisms inside it. We can mention the links between special centers in the brain and the eye. How did this complex system creation come about? According to biologists, the first step in the emergence of the eye during the evolutionary process was taken with the appearance of a small, light-sensitive area on the skin of some primitive living things. But what advantage could such a minute development on its own confer on a living thing in natural selection? As well as this, there needs to be a visual center formed in the brain and a nerve system linked to it. As long as these rather complicated mechanisms are not linked to one another, then we cannot expect what we call sight to emerge. Darwin believed that variations emerged by chance. If that were the case, would not the appearance of all the many variations that sight requires in various places in the organism at the same time and their working together turn into a mystical puzzle? However, a number of complementary changes working together in harmony and cooperation are needed for sight. Some mollusks' eyes have retina, cornea, and a lens of cellulose tissue just like ours. Now, how can we explain the evolutionary processes of these two very different types requiring a string of chance events just by natural selection? It is a matter for debate whether Darwinists have been able to provide a satisfactory answer to this question. 345 This problem is so great from the evolutionist point of view that the closer we look at the details, the worse the quandary the theory finds itself in. One important detail which needs to be looked at is the claim about the cell which came to be sensitive to light. Darwinists gloss this over by saying, sight may have started by a single cell becoming sensitive to light. But what kind of design is such a structure supposed to have had? The chemistry of sight in his book Darwin's Black Box, Michael stresses that the structure of the living cell and all other biochemical systems were unknown black boxes for Darwin and his contemporaries. Darwin assumed that these black boxes possessed very simple structures and could have come about by chance. Now, however, modern biochemistry has opened up these black boxes and revealed the irreducibly complex structure of life. States that Darwin's comments on the emergence of the eye seem convincing because of the primitive level of 19th-century science. Darwin persuaded much of the world that a modern eye evolved gradually from a simpler structure, but he did not even try to explain where his starting point, the relatively simple light-sensitive spot, came from. On the contrary, Darwin dismissed the question of the eye's ultimate origin. He had an excellent reason for declining the question. It was completely beyond 19th-century science. How the eye works, that is, what happens when a photon of light first hits the retina, simply could not be answered at that time. 346 So, how does the system, which Darwin glossed over as a simple structure, actually work? How do the cells in the eye's retinal layer perceive the light rays that fall on them? The answer to that question is rather complicated. When photons hit the cells of the retina, they activate a chain action, rather like a domino effect. The first of these domino pieces is a molecule called the Levin's retinal that is sensitive to photons. When struck by a photon, this molecule changes shape, which in turn changes the shape of a protein called rhodopsin to which it is tightly bound. Rhodopsin then takes a form that enables it to stick to another resident protein in the cell called transducin. Prior to reacting with rhodopsin, transducin is bound to another molecule called GDP. When it connects with rhodopsin, transducin releases the GDP molecule and is linked to a new molecule called GTP. That is why the new complex consisting of the two proteins, rhodopsin and transducin, and a smaller molecule, GTP, is called GTP transducin rhodopsin. But the process has only just begun. The new GTP transducin rhodopsin complex can now very quickly bind to another protein resident in the cell called phosphodiesterase. This enables the phosphodiesterase protein to cut yet another molecule resident in the cell, called cGMP. Since this process takes place in the millions of proteins in the cell, the cGMP concentration is suddenly decreased. How does all this help with sight? The last element of this chain reaction supplies the answer. The fall in the cGMP amount affects the ion channels in the cell. The so-called ion channel is a structure composed of proteins that regulate the number of sodium ions within the cell. Under normal conditions, the ion channel allows sodium ions to flow into the cell while another molecule disposes of the excess ions to maintain a balance. When the number of cGMP molecules falls, so does the number of sodium ions. This leads to an imbalance of charge across the membrane, which stimulates the nerve cells connected to these cells, forming what we refer to as an electrical impulse. Nerves carry the impulses to the brain and seeing happens there. 347 In brief, a single photon hits a single cell, and through a series of chain reactions the cell produces an electrical impulse. This stimulus is modulated by the energy of the photon, that is, the brightness of the light. Another fascinating fact is that all of the processes described so far happen in no more than 1/1000th of a second. As soon as this chain reaction is completed, other specialized proteins within the cells convert elements such as 11-cis-retinol, rhodopsin and transducin back to their original states. The eye is under a constant shower of photons, and the chain reactions within the eye's sensitive cells enable it to perceive each one of these. The process of sight is actually a great deal more complicated than the outline presented here would indicate. However, even this brief overview is sufficient to demonstrate the extraordinary nature of the system. There is such a complex, finely calculated system inside the eye that it is nonsensical to claim that it could have come about by chance. The system possesses a totally irreducibly complex structure. If even one of the many molecular parts that enter into chain reaction with each other were missing, or did not possess a suitable structure, then the system would not function at all. It is clear that the system deals a heavy blow to Darwin's explanation of life by chance. Michael Bim makes this comment on the chemistry of the eye and the theory of evolution. Now that the black box of vision has been opened, it is no longer enough for an evolutionary explanation of that power to consider only the anatomical structures of whole eyes, as Darwin did in the 19th century, and as popularizers of evolution continue to do today. Each of the anatomical steps and structures that Darwin thought were so simple actually involves staggeringly complicated biochemical processes that cannot be papered over with rhetoric. 348 The irreducibly complex structure of the eye not only definitively disproves the Darwinist theory, but also shows that life was created by the all-wise and all-powerful Allah. The lobster eye. There are many different types of eye in the living world. We are accustomed to the camera-type eye found in vertebrates. This structure works on the principle of the refraction of light, which falls onto the lens and is focused on a point behind the lens inside the interior of the eye. However, the eyes possessed by other creatures work by very different methods. One example is the lobster. A lobster's eye works on a principle of reflection, rather than that of refraction. The most outstanding characteristic of the lobster eye is its surface, which is composed of numerous squares. As shown in the picture, these squares are positioned most precisely. As one astronomer commented in Science, the lobster is the most unrectangular animal I've ever seen. But under the microscope a lobster's eye looks like perfect graph paper. 349 These well-arranged squares are in fact the ends of tiny square tubes forming a structure resembling a honeycomb. At first glance, the honeycomb appears to be made up of hexagons, although these are actually the front faces of hexagonal prisms. In the lobster's eye, there are the squares in place of hexagons. Even more intriguing is that the sides of each one of these square tubes are like mirrors that reflect the incoming light. This reflected light is focused onto the retina flawlessly. The sides of the tubes inside the eye are lodged at such perfect angles that they all focus onto a single point. The extraordinary nature of this system is quite indisputable. All of these perfect square tubes have a layer that works just like a mirror. Furthermore, each one of these cells is sited by means of precise geometrical alignments, so that they all focus the light at a single point. Michael Land, a scientist and researcher at the University of Sussex in England, was the first to examine the lobster eye structure in detail. Land stated that the eye had the most surprising structure. 350 It is obvious that the lobster eye presents a great difficulty for the theory of evolution. Most importantly, it exemplifies the concept of irreducible complexity. If even one of its features such as the facets of the eye, which are perfect squares, the mirrored sides of each unit, or the retina layer at the back were eliminated, the eye could never function. Therefore, it is impossible to maintain that the eye evolved step by step. It is scientifically unjustifiable to argue that such a perfect structure as this could have come about haphazardly. It is quite clear that the lobster eye was created by Allah as a miraculous system. One can find further traits in the lobster's eye that nullify the assertions of evolutionists. An interesting fact emerges when one looks at creatures with similar eye structures. The reflecting eye, of which the lobster's eye is one example, is found in only one group of crustaceans, the so-called long-bodied decapods. This family includes the lobsters, the prawns and shrimp. The other members of the crustacea class display the refracting type eye structure, which works on completely different principles from those of the reflecting type. Here, the eye is made up of hundreds of cells like a honeycomb. Unlike the square cells in a lobster eye, these cells are either hexagonal or round. Furthermore, instead of reflecting light, small lenses in the cells refract the light onto the focus on the retina. The majority of crustaceans have the refracting eye structure. According to evolutionist assumptions, all the creatures within the class Crustacea should have evolved from the same ancestor. Therefore, evolutionists claim that reflecting mirrored eye evolved from a refracting eye. However, such reasoning is impossible, because both eye structures function perfectly within their own systems and have no room for any transitional phase. A crustacean would be left sightless and would be eliminated by natural selection if the refracting lens in its eye were to diminish and be replaced by reflecting mirrored surfaces. It is, therefore, certain that both of these eye structures were created separately. There is such superb geometric precision in these eyes that believing that they came into being by chance is simply ludicrous. The creation of the ear. Another interesting example of the irreducibly complex organs in living things is the human ear. As is commonly known, the hearing process begins with vibrations in the air. These vibrations are enhanced in the external ear. Research has shown that that part of the external ear known as the concha works as a kind of megaphone, and sound waves are intensified in the external auditory canal. In this way, the volume of sound waves increases considerably. Sound intensified in this way enters the external auditory canal. This is the area from the external ear to the eardrum. One interesting feature of the auditory canal, which is some 3.5 centimeters long, is the wax it constantly secretes. This liquid contains an antiseptic property which keeps bacteria and insects out. Furthermore, the cells on the surface of the auditory canal are aligned in a spiral form directed towards the outside, so that the wax always flows towards the outside of the ear as it is secreted. Sound vibrations which pass down the auditory canal in this way reach the eardrum. This membrane is so sensitive that it can even perceive vibrations on the molecular level. By means of the exquisite sensitivity of the eardrum, you can easily hear somebody whispering from yards away. Or you can hear the vibration set up as you slowly rub two fingers together. Another extraordinary feature of the eardrum is that after receiving a vibration it returns to its normal state. Calculations have revealed that, after perceiving the tiniest vibrations, the eardrum becomes motionless again within up to 4/1000ths of a second. If it did not become motionless again so quickly, every sound we hear would echo in our ears. The eardrum amplifies the vibrations which come to it, and sends them onto the middle ear region. Here, there are three bones in an extremely sensitive equilibrium with each other. These three bones are known as the hammer, the anvil and the stirrup. Their function is to amplify the vibrations that reach them from the eardrum. But the middle ear also possesses a kind of buffer, to reduce exceedingly high levels of sound. This feature is provided by two of the body's smallest muscles, which control the hammer, anvil and stirrup bones. These muscles enable exceptionally loud noises to be reduced before they reach the inner ear. As a result of this mechanism, we hear sounds that are loud enough to shock the system at a reduced volume. These muscles are involuntary, and come into operation automatically, in such a way that even if we are asleep and there is a loud noise beside us, these muscles immediately contract and reduce the intensity of the vibration reaching the inner ear. The middle ear, which is so flawless, needs to maintain an important equilibrium. The air pressure inside the middle ear has to be the same as that beyond the eardrum, in other words, the same as the atmospheric air pressure. But this balance has been thought of, and a canal between the middle ear and the outside world which allows an exchange of air has been built in. This canal is the Eustachian tube, a hollow tube running from the inner ear to the oral cavity. The inner ear. It will be seen that all we have examined so far consists of the vibrations in the outer and middle ear. The vibrations are constantly passed forward, but so far there is still nothing apart from a mechanical motion. In other words, there is as yet no sound. The process whereby these mechanical motions begin to be turned into sound begins in the area known as the inner ear. In the inner ear is a spiral-shaped organ filled with the liquid. This organ is called the cochlea. The last part of the middle ear is the stirrup bone, which is linked to the cochlea by a membrane. The mechanical vibrations in the middle ear are sent onto the liquid in the inner ear by this connection. The vibrations which reach the liquid in the inner ear set up wave effects in the liquid. The inner walls of the cochlea are lined with small hair-like structures, called stereocilia, which are affected by this wave effect. These tiny hairs move strictly in accordance with the motion of the liquid. If a loud noise is emitted, then more hairs bend in a more powerful way. Every different frequency in the outside world sets up different effects in the hairs. But what is the meaning of this movement of the hairs? What can the movement of the tiny hairs in the cochlea and the inner ear have to do with listening to a concert of classical music, recognizing a friend's voice, hearing the sound of a car, or distinguishing the millions of other kinds of sounds? The answer is most interesting, and once more reveals the complexity of the ear. Each of the tiny hairs covering the inner walls of the cochlea is actually a mechanism which lies on top of 16,000 hair cells. When these hairs sense a vibration, they move and push each other, just like dominoes. This motion opens channels in the membranes of the cells lying beneath the hairs. And this allows the inflow of ions into the cells. When the hairs move in the opposite direction, these channels close again. Thus, this constant motion of the hairs causes constant changes in the chemical balance within the underlying cells, which in turn enables them to produce electrical signals. These electrical signals are forwarded to the brain by nerves, and the brain then processes them, turning them into sound. Science has not been able to explain all the technical details of this system. While producing these electrical signals, the cells in the inner ear also manage to transmit the frequencies, strengths, and rhythms coming from the outside. This is such a complicated process that science has so far been unable to determine whether the frequency distinguishing system takes place in the inner ear or in the brain. At this point, there is an interesting fact we have to consider concerning the motion of the tiny hairs on the cells of the inner ear. Earlier, we said that the hairs wave back and forth, pushing each other like dominoes. But usually the motion of these tiny hairs is very small. Research has shown that a hair motion of just by the width of an atom can be enough to set off the reaction in the cell. Experts who have studied the matter give a very interesting example to describe the sensitivity of these hairs. If we imagine a hair as being as tall as the Eiffel Tower, the effect on the cell attached to it begins with a motion equivalent to just 3 centimeters of the top of the tower. 351. Just as interesting is the question of how often these tiny hairs can move in a second. This changes according to the frequency of the sound. As the frequency gets higher, The number of times these tiny hairs can move reaches very high levels. For instance, a sound of a frequency of 20,000 causes these tiny hairs to move 20,000 times a second. Everything we have examined so far has shown us that the ear possesses an extraordinary structure. On closer examination, it becomes evident that the structure is irreducibly complex, since, in order for hearing to happen, it is necessary for all the component parts of the auditory system to be present and in complete working order. Take away any one of these, for instance, the hammer bone in the middle ear, or damage its structure, and you will no longer be able to hear anything. In order for you to hear, such different elements as the eardrum, the hammer, anvil and stirrup bones, the inner ear membrane, the cochlea, the liquid inside the cochlea, the tiny hairs that transmit the vibrations from the liquid to the underlying sensory cells, the latter cells themselves, the nerve network running from them to the brain, and the hearing center in the brain must all exist in complete working order. The system cannot develop by stages, because the intermediate stages would serve no purpose. Evolutionist theories regarding the origin of the ear. The irreducibly complex system in the ear is something that evolutionists can never satisfactorily explain. When we look at the theories evolutionists occasionally propose, we are met by a facile and superficial logic. For example, the writer Wiesel Ataman, who translated the book Im Anfang war der Wasserstoff, in the beginning was hydrogen, by the German biologist Hoyamar von Ditfurth, into Turkish, and who has come to be regarded as an evolution expert by the Turkish media, sums up his scientific theory on the origin of the ear and the so-called evidence for it in this way: our hearing organ, the ear, emerged as a result of the evolution of the endoderm and exoderm layers, which we call the skin. One proof of this is that we feel low sounds in the skin of our stomachs. 352 In other words, Ataman thinks that the ear evolved from the ordinary skin in other parts of our bodies, and sees our feeling low sounds in our skin as a proof of this. Let us first take Ataman's theory, and then the so-called proof he offers. We have just seen that the ear is a complex structure made up of dozens of different parts. To propose that this structure emerged with the evolution of layers of skin is, in a word, to build castles in the air. What mutation or natural selection effect could enable such an evolution to happen? Which part of the ear formed first? How could that part, the product of coincidence, have been chosen through natural selection even though it had no function? How did chance bring about all the sensitive mechanical balances in the ear, the eardrum, the hammer, anvil and stirrup bones, the muscles that control them, the inner ear, the cochlea, the liquid in it, the tiny hairs, the movement-sensitive cells, their nerve connections, etc.? There is no answer to these questions. In fact, to suggest that all this complex structure is just chance is actually an attack on human intelligence. However, in Michael Denton's words, to the Darwinist the idea is accepted without a ripple of doubt, the paradigm takes precedence. 353 Beyond the mechanisms of natural selection and mutation, evolutionists really believe in a magic wand that brings about the most complex systems by chance. The proof that Ataman supplies for this imaginary theory is even more interesting. He says, our feeling low sounds in our skin is proof. What we call sound actually consists of vibrations in the air. Since vibrations are a physical effect, of course they can be perceived by our sense of touch. For that reason it is quite normal that we should be able to feel high and low sounds physically. Furthermore, these sounds also affect bodies physically. The breaking of glass in a room under high intensities of sound is one example of this. The interesting thing is that the evolutionist writer Ataman should think that these effects are a proof of the evolution of the ear. The logic Ataman employs is the following: the ear perceives sound waves, our skin is affected by these vibrations, therefore, the ear evolved from the skin. Following Ataman's logic, one could also say, the ear perceives sound waves, glass is also affected by these, therefore the ear evolved from glass. Once one has left the bounds of reason, there is no theory that cannot be proposed. Other scenarios that evolutionists put forward regarding the origin of the ear are surprisingly inconsistent. Evolutionists claim that all mammals, including human beings, evolved from reptiles. But, as we saw earlier, reptiles' ear structures are very different from those of mammals. All mammals possess the middle ear structure made up of the 3 bones that have just been described, whereas there is only 1 bone in the middle ear of all reptiles. In response to this, evolutionists claim that 4 separate bones in the jaws of reptiles changed place by chance and migrated to the middle ear, and that again by chance they took on just the right shape to turn into the anvil and stirrup bones. According to this imaginary scenario, the single bone in reptiles' middle ears changed shape and turned into the hammer bone, and the exceedingly sensitive equilibrium between the 3 bones in the middle ear was established by chance. 354 This fantastical claim, based on no scientific discovery at all, it corresponds to nothing in the fossil record, is exceedingly self-contradictory. The most important point here is that such an imaginary change would leave a creature deaf. Naturally, a living thing cannot continue hearing if its jawbones slowly start entering its inner ear. Such a species would be at a disadvantage compared to other living things and would be eliminated, according to what evolutionists themselves believe. On the other hand, a living thing whose jawbones were moving towards its ear would end up with a defective jaw. Such a creature's ability to chew would greatly decrease, and even disappear totally. This, too, would disadvantage the creature, and result in its elimination. In short, the results which emerge when one examines the structure of ears and their origins clearly invalidate evolutionist assumptions. The Grolier Encyclopedia, an evolutionist source, makes the admission that the origin of the ear is shrouded in uncertainty. 355 Actually, anyone who studies the system in the ear with common sense can easily see that it is the product of Allah's magnificent creation. The reproduction of Rheobatraeus' irreducible complexity is not a feature that we only see at the biochemical level or in complicated organs. Many biological systems possessed by living things are irreducibly complex, and invalidate the theory of evolution for that reason. The extraordinary reproductive method of Rheobatraeus, a species of frog living in Australia, is an example of this. The females of this species use a fascinating method to protect their eggs after fertilization. They swallow them. The tadpoles remain and grow in the stomach for the first 6 weeks after they hatch. How is it possible that they can remain in their mother's stomach that long without being digested? A flawless system has been created to enable them to do so. First, the female gives up eating and drinking for those 6 weeks, which means the stomach is reserved solely for the tadpoles. However, another danger is the regular release of hydrochloric acid and pepsin in the stomach. These chemicals would normally quickly kill the offspring. However, this is prevented by a very special measure. The fluids in the stomach of the mother are neutralized by the hormone-like substance prostaglandin E2, which is secreted first by the egg capsules and then by the tadpoles. Hence, the offspring grow healthily, even though they are swimming in a pool of acid. How do the tadpoles feed inside the empty stomach? The solution to this has been provided, too. The eggs of this species are significantly larger than those of others, as they contain a yolk very rich in proteins, sufficient to feed the tadpoles for 6 weeks. The time of birth is arranged perfectly, as well. The esophagus of the female frog dilates during birth, like the dilation in mammals during delivery. Once the young have emerged, the esophagus and the stomach both return to normal, and the female starts feeding again. 356 The miraculous reproduction system of Rheobatraeus explicitly invalidates the theory of evolution, since the whole system is irreducibly complex. Every step has to take place fully in order for the frogs to survive. The mother has to swallow the eggs, and has to stop feeding completely for 6 weeks. The eggs have to release a hormone-like substance to neutralize stomach acids. The addition of the extra protein-rich yolk to the egg is another necessity. The widening of the female's esophagus cannot be coincidental. If all these things fail to happen in the requisite sequence, the fraglets would not survive, and the species would face extinction. Therefore, this system cannot have developed step by step, as asserted by the theory of evolution. The species has existed with this entire system intact since its first member came into existence. Another way of putting it is, they were created. Conclusion: in this section we have only examined a few examples of the concept of irreducible complexity. In fact, most organs and systems in living things possess the feature. On the biochemical level in particular, systems function by the working together of a number of independent parts, and cannot by any means be reduced to further simplicity. This fact invalidates Darwinism, which tries to account for the marvelous features of life by coincidental processes. Darwin said that if it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down. Today, modern biology has revealed countless examples of this. One can only conclude, then, that Darwinism has absolutely broken down. Chapter 13. Evolution and Thermodynamics. The second law of thermodynamics, which is accepted as one of the basic laws of physics, holds that under normal conditions all systems left on their own tend to become disordered, dispersed, and corrupted in direct relation to the amount of time that passes. Everything, whether living or not, wears out, deteriorates, decays, disintegrates, and is destroyed. This is the absolute end that all beings will face one way or another, and according to the law, the process cannot be avoided. This is something that all of us have observed. For example, if you take a car to a desert and leave it there, you would hardly expect to find it in a better condition when you came back years later. On the contrary, you would see that its tires had gone flat, its windows had been broken, its chassis had rusted, and its engine had stopped working. The same inevitable process holds true for living things. The second law of thermodynamics is the means by which this natural process is defined, with physical equations and calculations. This famous law of physics is also known as the law of entropy. In physics, entropy is the measure of the disorder of a system. A system's entropy increases as it moves from an ordered, organized, and planned state towards a more disordered, dispersed, and unplanned one. The more disorder there is in a system, the higher its entropy is. The law of entropy holds that the entire universe is unavoidably proceeding towards a more disordered, unplanned, and disorganized state. The truth of the second law of thermodynamics, or the law of entropy, has been experimentally and theoretically established. All foremost scientists agree that the law of entropy will remain the principal paradigm for the foreseeable future. Albert Einstein, the greatest scientist of our age, described it as the premier law of all of science. Sir Arthur Eddington also referred to it as the supreme metaphysical law of the entire universe. 357 Evolutionary theory ignores this fundamental law of physics. The mechanism offered by the theory of evolution totally contradicts the second law. The theory of evolution says that disordered, dispersed, and lifeless atoms and molecules spontaneously came together over time, in a particular order, to form extremely complex molecules such as proteins, DNA, and RNA, whereupon millions of different living species with even more complex structures gradually emerged. According to the theory of evolution, this supposed process, which yields a more planned, more ordered, more complex and more organized structure at each stage, was formed all by itself under natural conditions. The law of entropy makes it clear that this so-called natural process utterly contradicts the laws of physics. Evolutionist scientists are also aware of this fact. J. H. Rush states, in the complex course of its evolution, life exhibits a remarkable contrast to the tendency expressed in the second law of thermodynamics. Where the second law expresses an irreversible progression toward increased entropy and disorder, life evolves continually higher levels of order. 358 The evolutionist author Roger Lewin expresses the thermodynamic impasse of evolution in an article in Science, One problem biologists have faced is the apparent contradiction by evolution of the second law of thermodynamics. Systems should decay through time, giving less, not more, order. 359 Another defender of the theory of evolution, George Stavropoulos, states the thermodynamic impossibility of the spontaneous formation of life and the impossibility of explaining the existence of complex living mechanisms by natural laws in the well-known evolutionist journal American Scientist, yet, under ordinary conditions, no complex organic molecule can ever form spontaneously, but will rather disintegrate, in agreement with the second law. Indeed, the more complex it is, the more unstable it will be, and the more assured, sooner or later, its disintegration. Photosynthesis and all life processes, and even life itself, cannot yet be understood in terms of thermodynamics or any other exact science, despite the use of confused or deliberately confusing language. 360 As we have seen, the evolution claim is completely at odds with the laws of physics. The second law of thermodynamics constitutes an insurmountable obstacle for the scenario of evolution, in terms of both science and logic. Unable to offer any scientific and consistent explanation to overcome this obstacle, evolutionists can only do so in their imagination. For instance, Jeremy Rifkin notes his belief that evolution overwhelms this law of physics with a magical power. The entropy law says that evolution dissipates the overall available energy for life on this planet. Our concept of evolution is the exact opposite. We believe that evolution somehow magically creates greater overall value and order on Earth. 361 These words well indicate that evolution is a dogmatic belief rather than a scientific thesis. The misconception about open systems Some proponents of evolution have recourse to an argument that the second law of thermodynamics holds true only for closed systems, and that open systems are beyond the scope of this law. This claim goes no further than being an attempt by some evolutionists to distort scientific facts that invalidate their theory. In fact, a large number of scientists openly state that this claim is invalid, and violates thermodynamics. One of these is the Harvard scientist John Ross, who also holds evolutionist views. He explains that these unrealistic claims contain an important scientific error in the following remarks in Chemical and Engineering News: There are no known violations of the second law of thermodynamics. Ordinarily, the second law is stated for isolated systems, but the second law applies equally well to open systems. There is somehow associated with the field of far from equilibrium phenomena the notion that the second law of thermodynamics fails for such systems. It is important to make sure that this error does not perpetuate itself. 362 An open system is a thermodynamic system in which energy and matter flow in and out. Evolutionists hold that the world is an open system, that it is constantly exposed to an energy flow from the sun, that the law of entropy does not apply to the world as a whole, and that ordered, complex living beings can be generated from disordered, simple, and inanimate structures. However, there is an obvious distortion here. The fact that a system has an energy inflow is not enough to make that system ordered. Specific mechanisms are needed to make the energy functional. For instance, a car needs an engine, a transmission system, and related control mechanisms to convert the energy in petrol to work. Without such an energy conversion system, the car will not be able to use the energy stored in petrol. The same thing applies in the case of life as well. It is true that life derives its energy from the sun. However, solar energy can only be converted into chemical energy by the complex energy conversion systems in living things, such as photosynthesis in plants and the digestive systems of humans and animals. No living thing can live without such energy conversion systems. Without an energy conversion system, the sun is nothing but a source of destructive energy that burns, parches, or melts. As can be seen, a thermodynamic system without an energy conversion mechanism of some sort is not advantageous for evolution, be it open or closed. No one asserts that such complex and conscious mechanisms could have existed in nature under the conditions of the primeval Earth. Indeed, The real problem confronting evolutionists is the question of how complex energy-converting mechanisms such as photosynthesis in plants, which cannot be duplicated even with modern technology, could have come into being on their own. The influx of solar energy into the world would be unable to bring about order on its own. Moreover, no matter how high the temperature may become, amino acids resist forming bonds in ordered sequences. Energy by itself is incapable of making amino acids form the much more complex molecules of proteins, or of making proteins form the much more complex and organized structures of cell organelles. Ilya Prigogine and the myth of the self-organization of matter quite aware that the second law of thermodynamics renders evolution impossible. Some evolutionist scientists have made speculative attempts to square the circle between the two, in order to be able to claim that evolution is possible. One person distinguished by his efforts to marry thermodynamics and evolution is the Belgian scientist Ilya Prigogine. Starting out from chaos theory, Prigogine proposed a number of hypotheses in which order develops from chaos, disorder. However, despite all his best efforts, he was unable to reconcile thermodynamics and evolution. In his studies, he tried to link irreversible physical processes to the evolutionist scenario on the origin of life, but he was unsuccessful. His books, which are completely theoretical and include a large number of mathematical propositions which cannot be implemented in real life and which there is no possibility of observing, have been criticized by scientists, recognized as experts in the fields of physics, chemistry and thermodynamics, as having no practical and concrete value. For instance, P. Hohenberg, a physicist regarded as an expert in the fields of statistical mechanics and pattern formation, and one of the authors of the book Review of Modern Physics, sets out his comments on Prigogine's studies in the May 1995 edition of Scientific American. I don't know of a single phenomenon his theory has explained. 363 and Michel Izai, a theoretical physicist from Wisconsin University, has this to say about the fact that Prigogine's studies have reached no firm conclusion or explanation. In the just under 500 pages of his self-organization in non-equilibrium systems, there are just 4 graphs of real-world data, and no comparison of any of his models with experimental results. Nor are his ideas about irreversibility at all connected to self-organization, except for their both being topics in statistical physics. 364 The studies in the physical field by the determinedly materialist Prigogine also had the intention of providing support for the theory of evolution, because, as we have seen in the preceding pages, the theory of evolution is in clear conflict with the entropy principle, that is, the second law of thermodynamics. The law of entropy, as we know, definitively states that when any organized and complex structure is left to natural conditions, then loss of organization, complexity and information will result. In opposition to this, the theory of evolution claims that unordered, scattered, and unconscious atoms and molecules came together and gave rise to living things with their organized systems. Prigogine determined to try to invent formulae that would make processes of this kind feasible. However, all these efforts resulted in nothing but a series of theoretical experiments. The two most known theories that emerged as a result of that aim were the theory of self-organization and the theory of dissipative structures. The first of these maintains that simple molecules can organize together to form complex living systems. The second claims that ordered, complex systems can emerge in unordered, high-entropy systems. But these have no other practical and scientific value than founding new, imaginary worlds for evolutionists. The fact that these theories of Prigogine explain nothing, and have produced no results, is admitted by many scientists. The well-known physicist Joel Kaiser writes, his supposed criteria for predicting the stability of far-from equilibrium dissipative structures fails except for states very near equilibrium. 365 The theoretical physicist Michel Izai has this to say on the subject. Second, he tried to push forward a rigorous and well-grounded study of pattern formation and self-organization almost before anyone else. He failed. 366 F. Eugene Yates, editor of Self-Organizing Systems, The Emergence of Order, sums up the criticisms directed at Prigogine by Daniel L. Stein and the Nobel Prize-winning scientist Philip W. Anderson, in an essay in that same journal. The authors, Anderson and Stein, compare symmetry breaking in thermodynamic equilibrium systems, leading to phase change, and in systems far from equilibrium, leading to dissipative structures. Thus, the authors do not believe that speculation about dissipative structures and their broken symmetries can, at present, be relevant to questions of the origin and persistence of life. 367 In short, Prigogine's theoretical studies are of no value in explaining the origin of life. The same authors make this comment about his theories. Contrary to statements in a number of books and articles in this field, we believe that there is no such theory, and it even may be that there are no such structures as they are implied to exist by Prigogine, Haken, and their collaborators. 368 In essence, experts in the subject state that none of the theses Prigogine put forward possess any truth or validity, and that structures of the kind he discusses, dissipative structures, may not even really exist. Prigogine's claims are considered in great detail in Jean Brimont's article Science of Chaos or Chaos and Science, which makes their invalidity clear. Despite the fact that Prigogine did not manage to find a way to support evolution, the mere fact that he took initiatives of this sort was enough for the evolutionists to accord him the very greatest respect. A large number of evolutionists have welcomed Prigogine's concept of self-organization with great hope and a superficial bias. Prigogine's imaginary theories and concepts have nevertheless convinced many people who do not know much about the subject that evolution has resolved the dilemma of thermodynamics, whereas even Prigogine himself has accepted that the theories he has produced for the molecular level do not apply to living systems. For instance, a living cell, the problem of biological order involves the transition from the molecular activity to the supramolecular order of the cell. This problem is far from being solved. 369 These are the speculations that evolutionists have indulged in, encouraged by Prigogine's theories, which were meant to resolve the conflict between evolution and other physical laws. The difference between organized and ordered systems. If we look carefully at Prigogine and other evolutionists' claims, we can see that they have fallen into a very important trap. In order to make evolution fit in with thermodynamics, evolutionists are constantly trying to prove that a given order can emerge from open systems. And here it is important to bring out two key concepts to reveal the deceptive methods the evolutionists use. That deception lies in the deliberate confusing of two distinct concepts, ordered and organized. We can make this clear with an example. Imagine a completely flat beach on the seashore. When a strong wave hits the beach, mounds of sand, large and small, form bumps on the surface of the sand. This is a process of ordering. The seashore is an open system, and the energy flow, the wave, that enters it can form simple patterns in the sand, which look completely regular. From the thermodynamic point of view, it can set up order here where before there was none. But we must make it clear that those same waves cannot build a castle on the beach. If we see a castle there, we are in no doubt that someone has constructed it, because the castle is an organized system. In other words, it possesses a clear design and information. Every part of it has been made by an intelligent entity in a planned manner. The difference between the sand and the castle is that the former is an organized complexity, whereas the latter possesses only order, brought about by simple repetitions. The order formed from repetitions is as if an object, in other words the flow of energy entering the system, had fallen on the letter on a typewriter keyboard, writing hundreds of times. But the string of A's in an order repeated in this manner contains no information, and no complexity. In order to write a complex chain of letters actually containing information, in other words a meaningful sentence, paragraph or book, the presence of intelligence is essential. The same thing applies when a gust of wind blows into a dusty room. When the wind blows in, the dust which had been lying in an even layer may gather in one corner of the room. This is also a more ordered situation than that which existed before, in the thermodynamic sense, but the individual specks of dust cannot form a portrait of someone on the floor in an organized manner. This means that complex, organized systems can never come about as a result of natural processes. Although simple examples of order can happen from time to time, these cannot go beyond certain limits. But evolutionists point to this self-ordering which emerges through natural processes as a most important proof of evolution, portray such cases as examples of self-organization. As a result of this confusion of concepts, they propose that living systems could develop of their own accord from occurrences in nature and chemical reactions. The methods and studies employed by Prigogine and his followers, which we considered above, are based on this deceptive logic. However, as we made clear at the outset, organized systems are completely different structures from ordered ones. While ordered systems contain structures formed of simple repetitions, organized systems contain highly complex structures and processes, one often embedded inside the other. In order for such structures to come into existence, there is a need for intelligence, knowledge, and planning. Jeffrey Wicken, an evolutionist scientist, describes the important difference between these two concepts in this way: organized systems are to be carefully distinguished from ordered systems. Neither kind of system is random, but whereas ordered systems are generated according to simple algorithms and therefore lack complexity, organized systems must be assembled element by element according to an external wiring diagram with a high information content. Organization, then, is functional complexity and carries information. 370 Prigogine may be as a result of evolutionist wishful thinking resorted to a confusion of these two concepts, and advertised examples of molecules which ordered themselves under the influence of energy inflows as self-organization. The American scientists Charles B. Thaxton, Walter L. Bradley and Roger L. Olson, in their book titled The Mystery of Life's Origin, explain this fact as follows. In each case, random movements of molecules in a fluid are spontaneously replaced by a highly ordered behavior. Prigogine, Eigen, and others have suggested that a similar sort of self-organization may be intrinsic in organic chemistry and can potentially account for the highly complex macromolecules essential for living systems. But such analogies have scant relevance to the origin of life question. A major reason is that they fail to distinguish between order and complexity. 371 And this is how the same scientists explain the logical shallowness and distortion of claiming that water turning into ice is an example of how biological order can spontaneously emerge. It has often been argued by analogy to water crystallizing to ice that simple monomers may polymerize into complex molecules such as protein and DNA. The analogy is clearly inappropriate, however. The atomic bonding forces draw water molecules into an orderly crystalline array when the thermal agitation, or entropy driving force, is made sufficiently small by lowering the temperature. Organic monomers such as amino acids resist combining at all at any temperature, however, much less some orderly arrangement. 372 Prigogine devoted his whole career to reconciling evolution and thermodynamics. But even he admitted that there was no resemblance between the crystallization of water and the emergence of complex biological structures. The point is that in a non-isolated system there exists a possibility for a formation of ordered, low-entropy structures at sufficiently low temperatures. This ordering principle is responsible for the appearance of ordered structures such as crystals as well as for the phenomena of phase transitions. Unfortunately, this principle cannot explain the formation of biological structures. 373 In short, no chemical or physical effect can explain the origin of life, and the concept of the self-organization of matter will remain a fantasy. Self-organization, a materialist dogma. The claim that evolutionists maintain with the concept of self-organization is the belief that inanimate matter can organize itself and generate a complex living thing. This is an utterly unscientific conviction. Observation and experiment have incontrovertibly proven that matter has no such property. The famous English astronomer and mathematician Professor Fred Hoyle notes that matter cannot generate life by itself, without deliberate interference. If there were a basic principle of matter which somehow drove organic systems toward life, its existence should easily be demonstrable in the laboratory. One could, for instance, take a swimming bath to represent the primordial soup. Fill it with any chemicals of a non-biological nature you please. Pump any gases over it, or through it, you please, and shine any kind of radiation on it that takes your fancy. Let the experiment proceed for a year and see how many of those 2,000 enzymes, proteins produced by living cells, have appeared in the bath. I will give the answer, and so save the time and trouble and expense of actually doing the experiment. You will find nothing at all, except possibly for a tarry sludge composed of amino acids and other simple organic chemicals. 374 Evolutionary biologist Andrew Scott admits the same fact: take some matter, heat while stirring and wait. That is the modern version of Genesis. The fundamental forces of gravity, electromagnetism and the strong and weak nuclear forces are presumed to have done the rest. But how much of this neat tale is firmly established, and how much remains hopeful speculation? In truth, the mechanism of almost every major step, from chemical precursors up to the first recognizable cells, is the subject of either controversy or complete bewilderment. 375 So why do evolutionists continue to believe in scenarios such as the self-organization of matter, which have no scientific foundation? Why are they so determined to reject the intelligence and signs of creation that can so clearly be seen in living systems? The answer to these questions lies hidden in the materialist philosophy that the theory of evolution is fundamentally constructed on. Materialist philosophy believes that only matter exists, for which reason living things need to be accounted for in a manner based on matter. It was this difficulty which gave birth to the theory of evolution, and no matter how much it conflicts with the scientific evidence, it is defended for just that reason. A professor of chemistry from New York University and DNA expert, Robert Shapiro, explains this belief of evolutionists about the self-organization of matter and the materialist dogma lying at its heart as follows: another evolutionary principle is therefore needed to take us across the gap from mixtures of simple natural chemicals to the first effective replicator. This principle has not yet been described in detail or demonstrated, but it is anticipated, and given names such as chemical evolution and self-organization of matter. The existence of the principle is taken for granted in the philosophy of dialectical materialism, as applied to the origin of life by Alexander Oparin. 376 The truths that we have been examining in this section clearly demonstrate the impossibility of evolution in the face of the second law of thermodynamics. The concept of self-organization is another dogma that evolutionist scientists are trying to keep alive despite all the scientific evidence. Chapter 14 Information Theory and the End of Materialism. Materialist philosophy lies at the basis of the theory of evolution. Materialism rests on the supposition that everything that exists is matter. According to this philosophy, matter has existed since eternity, will continue to exist forever, and there is nothing but matter. In order to provide support for their claim, materialists use a logic called reductionism. This is the idea that things which are not observable can also be explained by material causes. To make matters clearer, let us take the example of the human mind. It is evident that the mind cannot be touched or seen. Moreover, it has no center in the human brain. This situation unavoidably leads us to the conclusion that mind is a concept beyond matter. Therefore, the being which we refer to as I, who thinks, loves, fears, worries, and feels pleasure or pain, is not a material being in the same way as a sofa, a table or a stone. Materialists, however, claim that mind is reducible to matter. According to the materialist claim, thinking, loving, worrying and all our mental activities are nothing but chemical reactions taking place between the atoms in the brain. Loving someone is a chemical reaction in some cells in our brain, and fear is another. The famous materialist philosopher Karl Vogt is notorious for his assertion that the brain secretes thought just as the liver secretes bile. 377 Bile, however, is matter, whereas there is no evidence that thought is. Reductionism is a logical deduction. However, a logical deduction can be based on solid grounds or on shaky ones. For this reason, the question we need to ask is, what happens when reductionism is compared to scientific data? 19th-century materialist scientists and thinkers thought that the answer would be that science verifies reductionism. 20th-century science, however, has revealed a very different picture. One of the most salient features of this picture is information, which is present in nature and can never be reduced to matter. The difference between matter and information: we earlier mentioned that there is astonishingly comprehensive information contained in the DNA of living things. Something as small as 100,000th of a millimeter across contains a sort of databank that specifies all the physical details of the body of a living thing. Moreover, the body also contains a system that reads this information, interprets it and carries out production in line with it. In all living cells, the information in the DNA is read by various enzymes, and proteins are produced. This system makes possible the production of millions of proteins every second, of just the required type for just the places where they are needed in our bodies. In this way, dead eye cells are replaced by living ones, and old blood cells by new ones. At this point, let us consider the claim of materialism. Is it possible that the information in DNA could be reduced to matter, as materialists suggest? Or, in other words, can it be accepted that DNA is merely a collection of matter, and the information it contains came about as a result of the random interactions of such pieces of matter? All the scientific research, experiments and observations carried out in the 20th century show that the answer to this question is a definite no. The director of the German Federal Physics and Technology Institute, Professor Werner Gitt, has this to say on the issue: a coding system always entails a non-material intellectual process. A physical matter cannot produce an information code. All experiences show that every piece of creative information represents some mental effort and can be traced to a personal idea giver who exercised his own free will, and who is endowed with an intelligent mind. There is no known law of nature, no known process and no known sequence of events which can cause information to originate by itself in matter. 378 Werner Gitt's words summarize the conclusions of information theory, which has been developed in the last 50 years, and which is accepted as a part of thermodynamics. Information theory investigates the origin and nature of the information in the universe. The conclusion reached by information theoreticians as a result of long studies is that information is something different from matter. It can never be reduced to matter. The origin of information and physical matter must be investigated separately. For instance, let us think of the source of a book. A book consists of paper, ink, and the information it contains. Paper and ink are material elements. Their source is again matter. Paper is made of cellulose, and ink of various chemicals. However, the information in the book is non-material, and cannot have a material source. The source of the information in each book is the mind of the person who wrote it. Moreover, this mind determines how the paper and ink will be used. A book initially forms in the mind of the writer. The writer builds a chain of logic in his mind, and orders his sentences. As a second step, he puts them into material form, which is to say that he translates the information in his mind into letters, using a pen, a typewriter or a computer. Later, these letters are printed in a publishing house, and take the shape of a book made up of paper and ink. We can therefore state this general conclusion: if physical matter contains information, then there is a mind possessing superior knowledge that created that matter. It is the Almighty Allah who perfectly created the entire universe. The origin of the information in nature. When we apply this scientific definition of information to nature, a very important result ensues. This is because nature overflows with an immense body of information, as, for example, in the case of DNA, and since this information cannot be reduced to matter, it therefore comes from a source beyond matter. One of the foremost advocates of the theory of evolution, George C. Williams, admits this reality, which most materialists and evolutionists are reluctant to see. Williams has strongly defended materialism for years, but in an article he wrote in 1995, he states the incorrectness of the materialist, reductionist, approach which holds that everything is matter. Evolutionary biologists have failed to realize that they work with two more or less incommensurable domains, that of information and that of matter. These two domains will never be brought together in any kind of the sense usually implied by the term reductionism. The gene is a package of information, not an object. In biology, when you're talking about things like genes and genotypes and gene pools, you're talking about information, not physical objective reality. This dearth of shared descriptors makes matter and information two separate domains of existence, which have to be discussed separately, in their own terms. 379 Therefore, contrary to the supposition of materialists, the source of the information in nature cannot be matter itself. The source of information is not matter but a superior wisdom beyond matter. This wisdom existed prior to matter. The possessor of this wisdom is Allah, the Lord of all the worlds. Matter was brought into existence, given form, and organized by him. Gerald Schroeder, an MIT-trained Israeli scientist who worked in physics and biology and authored The Science of God, makes a number of important comments on this subject. In his more recent book, Science Reveals the Ultimate Truth, Schroeder sets out the conclusion revealed by such branches of science as molecular biology and quantum physics: a single consciousness, a universal wisdom, pervades the universe. The discoveries of science, those that search the quantum nature of subatomic matter, have moved us to the brink of a startling realization: all existence is the expression of this wisdom. In the laboratories we experience it as information that first physically articulated as energy and then condensed into the form of matter. Every particle, every being, from atom to human, appears to represent the level of information, of wisdom. 380 According to Schroeder, the contemporary scientific conclusions have enabled science and theology to agree on a common point. That is the fact of creation. Science has now reached the point of discovering this fact which the divine religions have been teaching for thousands of years. Materialist admissions We have already described how one of the fundamental principles that make up life is knowledge, and it is clear that this knowledge proves the existence of an intelligent creator. The theory of evolution, which tries to account for life as being the result of coincidences in a purely material world, and the materialist philosophy it is based on, are quite helpless in the face of this reality. When we look at evolutionists' writings, we sometimes see that this helplessness is openly admitted. One forthright authority on this subject is the well-known French zoologist Pierre-Paul Grass. He is a materialist and an evolutionist, although he sometimes openly admits the quandaries Darwinist theory faces. According to Grass, the most important truth which invalidates the Darwinist account is the knowledge that gives rise to life. Any living being possesses an enormous amount of intelligence, very much more than is necessary to build the most magnificent of cathedrals. Today, this intelligence is called information, but it is still the same thing. It is not programmed as in a computer, but rather it is condensed on a molecular scale in the chromosomal DNA or in that of every other organelle in each cell. This intelligence is the sine qua non of life. Where does it come from? This is a problem that concerns both biologists and philosophers, and, at present, science seems incapable of solving it. 381 The reason why Pierre-Paul Grass says, science seems incapable of solving it, is that he does not want any non-materialist explanation to be thought of as scientific. However, science itself invalidates the hypotheses of materialist philosophy, and proves the existence of a creator. Grass and other materialist scientists either ignore this reality, or else say, science does not explain this. They do this because they are materialists first and scientists second, and they continue to believe in materialism, even if science demonstrates the exact opposite. For this reason, In order to possess a correct scientific attitude, one has to distinguish between science and materialist philosophy. Chapter 15. Distinguishing between science and materialism. The information we have considered throughout this book has shown us that the theory of evolution has no scientific basis, and that, on the contrary, evolutionist claims conflict with scientific facts. In other words, the force that keeps evolution alive is not science. Evolution may be maintained by some scientists, but behind it there is another influence at work. This other influence is materialist philosophy. The theory of evolution is simply materialist philosophy applied to nature, and those who support that philosophy do so despite the scientific evidence. This relationship between materialism and the theory of evolution is accepted by authorities on these concepts. For example, that discovery of Darwin was described by Leon Trotsky as the highest triumph of the dialectic in the whole field of organic matter. 382 The evolutionary biologist Douglas Futema writes, together with Marx's materialist theory of history and society, Darwin hewed the final planks of the platform of mechanism and materialism. 383 And the evolutionary paleontologist Stephen Jay Gould says, Darwin applied a consistent philosophy of materialism to his interpretation of nature. 384 Materialist philosophy is one of the oldest beliefs in the world, and assumes the absolute and exclusive existence of matter as its basic principle. According to this view, matter has always existed, and everything that exists consists of matter. Materialism denies the evident existence of a creator. So the question becomes one of why the materialist point of view is false. One method of testing whether a philosophy is true or false is to investigate the claims it makes about science by using scientific methods. For instance, a philosopher in the 10th century could have claimed that there was a divine tree on the surface of the moon and that all living things actually grew on the branches of this huge tree like fruit, and then fell off onto the earth. Some people might have found this philosophy attractive and believed in it. But in the 21st century, at a time when man has managed to walk on the moon, it is no longer possible to seriously hold such a belief. Whether such a tree exists there or not can be determined by scientific methods, that is, by observation and experiment. We can therefore investigate by means of scientific methods the materialist claim that matter has existed for all eternity and that this matter can organize itself without a supramaterial creator and cause life to begin. When we do this, we see that materialism has already collapsed, because the idea that matter has existed since the beginning of time has been overthrown by the Big Bang theory, which shows that the universe was created from nothingness. The claim that matter organized itself and created life is the claim that we call the theory of evolution, which this book has been examining and which has been shown to have collapsed. However, if someone is determined to believe in materialism and puts his devotion to materialist philosophy before everything else, then he will act differently. If he is a materialist first and a scientist second, he will not abandon materialism when he sees that evolution is disproved by science. On the contrary, he will attempt to uphold and defend materialism by trying to support evolution, no matter what. This is exactly the predicament that evolutionists defending the theory of evolution find themselves in today. Interestingly enough, they also confess this fact from time to time. A well-known geneticist and outspoken evolutionist, Richard C. Lewontin from Harvard University, confesses that he is a materialist first and a scientist second in these words. It is not that the methods and institutions of science somehow compel us accept the material explanation of the phenomenal world, but, on the contrary, that we are forced by our a priori adherence to material causes to create an apparatus of investigation and a set of concepts that produce material explanations, no matter how counterintuitive, no matter how mystifying to the uninitiated. Moreover, that materialism is absolute, so we cannot allow a divine intervention. 393 Allow a divine foot in the door. 385 The term a priori that Lewontin uses here is quite important. This philosophical term refers to a presupposition not based on any experimental knowledge. A thought is a priori when you consider it to be correct and accept it as so even if there is no information available to confirm it. As the evolutionist Lewontin frankly states, materialism is an a priori commitment for evolutionists, who then try to adapt science to this preconception. Since materialism definitely necessitates denying the existence of a creator, they embrace the only alternative they have to hand, which is the theory of evolution. It does not matter to such scientists that evolution has been belied by scientific facts, because they have accepted it a priori as true. This prejudiced behavior leads evolutionists to a belief that unconscious matter composed itself, which is contrary not only to science, but also to reason. The concept of this self-organization of matter, which we examined in an earlier chapter, is an expression of this. Evolutionist propaganda, which we constantly come across in the Western media and in well-known and esteemed science magazines, is the outcome of this ideological necessity. Since evolution is considered to be indispensable, it has been turned into a taboo subject by the circles that set the standards of science. Some scientists find themselves in a position where they are forced to defend this far-fetched theory, or at least avoid uttering any word against it, in order to maintain their reputations. Academics in Western countries have to have articles published in certain scientific journals in order to attain and hold on to their professorships. All of the journals dealing with biology are under the control of evolutionists, and they do not allow any anti-evolutionist article to appear in them. Biologists, therefore, have to conduct their research under the domination of this theory. They, too, are part of the materialist order, which regards evolution as an ideological necessity, which is why they blindly defend all the impossible coincidences we have been examining in this book. The definition of the scientific cause the German biologist Hoymar von Ditfurth, a prominent evolutionist, is a good example of this bigoted materialist understanding. After Ditfurth cites an example of the extremely complex composition of life, this is what he says concerning the question of whether it could have emerged by chance or not. Is such a harmony that emerged only out of coincidences possible in reality? This is the basic question of the whole of biological evolution. Critically speaking, we can say that somebody who accepts the modern science of nature has no other alternative than to say yes, because he aims to explain natural phenomena by means that are understandable and tries to derive them from the laws of nature without reverting to supernatural interference. 386 Yes, as Ditfurth states, the materialist scientific approach adopts as its basic principle explaining life by denying supernatural interference, that is, creation. Once this principle is adopted, even the most impossible scenarios are easily accepted. It is possible to find examples of this dogmatic mentality in almost all evolutionist literature. Professor Ali Demirsoy, the well-known advocate of evolutionary theory in Turkey, is just one of many. According to Demirsoy, the probability of the coincidental formation of cytochrome C, an essential protein for life, is as unlikely as the possibility of a monkey writing the history of humanity on a typewriter without making any mistakes. 387 There is no doubt that to accept such a possibility is actually to reject the basic principles of reason and common sense. Even one single correctly formed letter written on a page makes it certain that it was written by a person. When one sees a book of world history, it becomes even more certain that the book has been written by an author. No logical person would agree that the letters in such a huge book could have been put together by chance. However, it is very interesting to see that the evolutionist scientist Professor Ali Demirsoy accepts this sort of irrational proposition. In essence, the probability of the formation of a cytochrome C sequence is as likely as zero. That is, if life requires a certain sequence, it can be said that this has a probability likely to be realized once in the whole universe. Otherwise some metaphysical powers beyond our definition must have acted in its formation. To accept the latter is not appropriate for the scientific cause. We thus have to look into the first hypothesis. 388 Demirsoy writes that he prefers the impossible, in order not to have to accept supernatural forces, in other words, the existence of a creator. However, The aim of science is not to avoid accepting the existence of supernatural forces. Science can get nowhere with such an aim. It should simply observe nature, free of all prejudices, and draw conclusions from these observations. If these results indicate that there is planning by a supernatural intelligence, which is the case in every corner of the universe, then science must accept the fact. Under close examination, what they call the scientific cause is actually the materialist dogma that only matter exists and that all of nature can be explained by material processes. This is not a scientific cause, or anything like it, it is just materialist philosophy. This philosophy hides behind such superficial words as scientific cause and obliges scientists to accept quite unscientific conclusions. Not surprisingly, when Demersoy cites another subject, the origins of the mitochondria in the cell, he openly accepts chance as an explanation, even though it is quite contrary to scientific thought. The heart of the problem is how the mitochondria have acquired this feature, because attaining this feature by chance, even by one individual, requires extreme probabilities that are incomprehensible. The enzymes providing respiration and functioning as a catalyst in each step in a different form make up the core of the mechanism. A cell has to contain this enzyme sequence completely, otherwise it is meaningless. Here, despite being contrary to biological thought, in order to avoid a more dogmatic explanation or speculation, we have to accept, though reluctantly, that all the respiration enzymes completely existed in the cell before the cell first came in contact with oxygen. 389 The conclusion to be drawn from such pronouncements is that evolution is not a theory arrived at through scientific investigation. On the contrary, the form and substance of this theory were dictated by the requirements of materialistic philosophy. It then turned into a belief or a dogma in spite of concrete scientific facts. Again, from evolutionist literature, we can clearly see that all of this effort has a purpose that requires maintaining, at no matter what cost, that living things were not created. Coming to terms with the shocks, as we recently stressed, materialism is the belief that categorically rejects the existence of the non-material, or the supernatural. Science, on the other hand, is under no obligation to accept such a dogma. The duty of science is to observe nature and produce results. And science does reveal the fact that living things were created. This is something demonstrated by scientific discoveries. When we examine the fantastically complex structures in living things, we see that they possess such extraordinary features that they can never be accounted for by natural processes and coincidences. Every instance of extraordinary feature is evidence for an intelligence that brought it into being. Therefore, we must conclude that life, too, was created by a power. This power belongs to a non-material wisdom, the superior wisdom of the all-powerful Allah, who rules all of nature. In short, life and all living things were created. This is not a dogmatic belief like materialism, but a plain fact revealed by scientific observation and experiment. We see that this fact comes as a terrible shock for scientists who are used to believing in materialism, and that materialism is a science. See how this shock is described by Michael Bay, one of the most important scientists to stand against the theory of evolution in the world today. The resulting realization that life was designed by an intelligence is a shock to us in the 20th century who have gotten used to thinking of life as the result of simple natural laws. But other centuries have had their shocks, and there is no reason to suppose that we should escape them. 390 Mankind has been freed from such dogmas as that the world is flat, or that it is the center of the universe. And it is now being freed from the materialist and evolutionist dogma that life came about by itself. The duty that befalls a true scientist in this respect is to do away with materialist dogma and evaluate the origin of life and living things with the honesty and objectivity befitting a real scientist. A real scientist must come to terms with the shock, and not tie himself to outdated 19th century dogmas and defend impossible scenarios. Conclusion: throughout this book we have examined the scientific evidence for the origin of life, and what emerges clearly demonstrates that life was not the result of chance, as claimed by Darwinism and materialist philosophy in general. Living species could not have evolved from one another through a string of coincidences. On the contrary, all living things were independently and flawlessly created. As the 21st century dawns, science offers but one answer to the question of the origin of life: creation. The important thing is that science has confirmed the truth which religion has been witness to from the dawn of history to the present day. Allah created the universe and all the living things in it from nothing. And it was Allah who created man from nothing and blessed him with countless characteristics. This truth has been sent down to man since the dawn of time by prophets, and revealed in holy books. Every prophet has told the communities to whom he was sent that Allah created man and all living things. The Bible and the Quran all tell of the news of creation in the same way. In the Quran, Allah announces in a number of verses that it was He who created the universe and all the living things in it from nothing, and flawlessly ordered them. In this verse, it is declared that creation belongs to Him, your Lord is Allah, who created the heavens and the earth in 6 days and then settled Himself firmly on the throne. He covers the day with the night, each pursuing the other urgently, and the sun and moon and stars are subservient to His command. Both creation and command belongs to Him. Blessed be Allah, the Lord of all the worlds. Surat Al-Araf, 54. Just as Allah created everything that exists, so He created the world we live in today, and made it capable of supporting life. This fact is revealed in certain verses. As for the earth, we stretched it out and cast firmly embedded mountains in it and made everything grow in due proportion on it. And we put livelihoods in it both for you and for those you do not provide for. Surat Al-Hijr, 1920, and the earth how we stretched it out and cast firmly embedded mountains onto it and caused luxuriant plants of every kind to grow in it. Surah Kahf, 7-8, the above verses announce that all plants were created by Allah. All plants, known and unknown, all trees, grasses, fruit, flowers, seaweed and vegetables were created by Allah. And the same thing applies to animals. All of the millions of different animal species that live, or have ever lived, on earth, were created by Allah. Fish, reptiles, birds, mammals, horses, giraffes, squirrels, deer, sparrows, eagles, dinosaurs, whales, and peacocks were all created from nothing by Allah, the Lord of infinite art and knowledge. Allah's creation of the different species of living things is mentioned in the verses. Allah created every animal from water. Some of them go on their bellies, some of them on 2 legs, and some on 4. Allah creates whatever He wills. Allah has power over all things. Surat An-Nur, 45, and He created livestock. There is warmth for you in them, and various uses and some you eat. Surat An-Nahl, 5, and Allah created man in exactly the same way. It is revealed in the Quran that Adam, the first man, was created from mud, and then all subsequent people came into existence from each other by a simple liquid, sperm. Furthermore, man had a soul breathed into him, unlike all the other species in the world. The Quran has this to say about the truth of the creation of man: He who has created all things in the best possible way. He commenced the creation of man from clay then produced his seed from an extract of base fluid. Surat Az-Sajda, 7-9, man's duty, as we made clear at the start. Science has once again revealed the truth of creation, as handed down in the Quran. Scientific discoveries show that living things possess extraordinary features, and that they were brought into existence by a superior intelligence and knowledge. Biological observations show that one living species cannot turn into another, and that for that reason, if one could go back in time, one would eventually come across, for each species, the first individuals that ever existed and that were created from nothing. For example, since eagles have always been eagles, if we could go back in time, we would arrive at the first pair, or group, of eagles who were created from nothing. In fact, the fossil record confirms this, and shows that different living species suddenly emerged with all their particular, individual features. These species may have been created at different points in time and settled in different parts of the world, but this all happened through the will of Allah. In short, Science reveals the proof we have considered that living things were all created by Allah. However, science goes no further than that. It is the Quran, the holy book that has come down to us from Allah, that introduces us to the essence of Allah and is the sole source of truth on every subject, that tells us why we were created and what the reason for our lives is. It is stated in the Quran that the reason for our creation is so that we might believe in Allah, our Lord, and serve Him. In one verse, He says, "I only created jinn and man to worship me." Surah Ad-Dariyat, 56. The duty falling to everyone who grasps the truth of creation is to live in accordance with that verse, and to say, "Why indeed should I not worship Him who brought me into being, Him to whom you will be returned?" Surah Yasin, 22. Like every believer, as described in the Quran. As for those who still deny Allah and the truth of creation, despite all the evidence before their eyes, their minds have been conquered by their own pride. One of Allah's holy verses describes how helpless and powerless these individuals really are, mankind. An example has been made, so listen to it carefully. Those whom you call upon besides Allah are not even able to create a single fly, even if they were to join together to do it. And if a fly steals something from them, they cannot get it back. How feeble are both the seeker and the sought. Surat Al-Hajj, 73.